That's when you know it's working. Okay, here we go. Special senses. This is a cool lecture. I love this lecture. Just so you know. What are the special senses? Anybody? Yeah? Name one.
Gustation.
Gustation. Yes. Taste.
Olfactory.
Have I told you my Gustav story?
Yes. Yeah.
Okay, go ahead. What's the next one? Olfaction, smell. Vision. Hearing.
And equilibrium.
Equilibrium. Okay. How many do you have? Well, there we go. We've already been through them. 5 of them. Olfaction, gustation, vision, equilibrium, and hearing. Let's start with the first one. These will go pretty fast. Some of these systems are pretty easy. Olfactory organs is how we smell. The olfactory organ is located in the nasal cavity above the upper concha. Okay, so it's in the very top of your nose. So it's probably, no matter how hard you put your finger up in your nose, you probably can't really get into it. It's on either side, obviously. It's made up of 2 layers. This is just basically the mucosa of that upper part. The olfactory epithelium has a bunch of receptors in it, and the lamina propria, which has a bunch of nerves running through it, which you'll see here in a second. The olfactory epithelium contains olfactory receptors. Now, what are olfactory receptors? They're not little things standing up there with their hands out. They're just little chemical receptors that are laying there in the mucosal wall. There are some supporting cells under it. There are some basal stem cells underneath it, because these cells are continually being replaced, as opposed to most cells in the many cells in the nervous system. We replace these. The lamina propria just below it is basically just some areolar tissue, blood vessels, nerves, and some olfactory glands. We'll talk about the olfactory glands a bit. So here's kind of a picture, which is kind of cool. So here's your lower, middle, and upper concha, and the area in question is up here above your upper concha. And notice that these olfactory nerve fibers are very short, and they go right through the old cribriform plate. Cribriform plate is part of what bone? Ethmoid bone. And it goes into this little bulby thing here, which is called the olfactory bulb. And the olfactory bulb then follows the olfactory tract. And the olfactory tract goes into the brain. Okay? Now, think about this for a minute. This is the only system in the brain that goes straight from receptor to cerebrum, your smell. It doesn't go to the thalamus. It doesn't go to any relay center. It doesn't go to any other nuclei. It just goes straight into the olfactory part of the brain. Now, also think about this. My father-in-law used to have a hunting dog. And I really thought it was bullshit. You know, I really did. We would go out and we would follow this dog through the woods, and we'd go up and down the hills, up and down the hills, up and down the hills, and all of a sudden the dog would go, and he would point at this bunch of weeds or something, you know, that were maybe 20 feet off. And Father would say, he's on point. Let's be quiet. You move to the right, Hal, I'll move to the left, and, you know, get your gun ready. And then he would say, okay, Leroy, that was the name of the dog. Leroy, move on in. Leroy would move on in. And then up would come about 20 quail. And it scared the shit out of me. They were so loud. And, you know, I think I killed 2 quail in my whole life. I was such a lousy shot. But I was just scared to death when those things would come up. But the point of the matter was, that dog could smell those birds from 50 feet away. And he'd be running just as fast as he could run, and then he'd go down on point. We had a squirrel dog, too. It was the same thing. You'd be tracing through the woods for an hour, and then all of a sudden you'd hear him way off in the distance going, "Wah, wah, 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 wah." You go over there, and there he is on a tree with his feet up on the tree, you know, and there's no way there's a squirrel up in that tree. And we would split up. He'd go one way around the tree, and I'd go other way around the tree. And here's this poor squirrel up there clinging to the side of the tree, trying to move around the tree so we wouldn't see him. And once we saw him, it was a pretty easy shot. I did kill a few squirrels in my life, but that all ended many, many, many years ago. Anyway, the point being is that most of the animals have far, far better noses than we have. And one of the reasons their noses are so good is because of this business about it doesn't go synapse anywhere. It just goes straight to the brain. And so for animals, and so many of them rely on their sense of smell to live, to hunt, and to find food, that the olfactory system is really pretty specialized and pretty incredible. I don't smell hardly anything. But there are people who work real hard at their sense of smell, and they smell all these different things, and they take wine and they swirl it around and say, oh, it's got a good nose, and it's so oaky, and it's, oh, yes, there's some caramel here, and I'm like, get out of here. It's a glass of wine. But anyway. All right, so here's the deal. We have all these things we smell, and we breathe them in, and they come up through our nose, and when they get up into here, little chemicals, they're the things that smell are actually chemicals that are floating in the air, and they land up here, and they attach to a receptor, and that receptor then gets activated and sends a message to the brain. Now, in this case, that receptor is a receptor for roses, and your brain has learned that. You've smelled a bunch of roses in your life, and so when you smell, that must be a rose. And so that's the way it works, because the receptors are all very specific for different things. So there's a specific receptor for sulfur gas. There's a specific receptor for any good smell and any bad smell you can think of. Okay? We have, I want to say, a couple of hundred different receptors up here. That means we could actually characterize that many different odors. So here's what it looks like under the microscope. This is the lamina propria back here. It's here's the surface out here. Here's the little particles that are coming in here and landing in this milieu of moisture up here that they're picked up. And these olfactory receptor cells, these are the actual cells that smell, have these little roots on them down here, these olfactory cilia that pick up these smells by basically, because the olfactory cilia contain receptor proteins. And I just want you to understand that this is a chemical process, okay? As we go through these different receptors later today, some of them are mechanical, like that's the way we hear. It's a mechanical thing. Things are actually moving. But this is a chemical thing. Same with our taste. It's determined by chemical receptors in the nose. Basal cell, you see they're regenerating cells up here all the time to replace worn out olfactory cells. We are continually burning up our olfactory cells and need to be replaced. This olfactory gland up here secretes this moist, this juice that's kind of a mucus layer that spreads out there that then the receptors can dissolve in, and once they dissolve in that surface mucus, then they can be they can attract and hook on to the receptors that are in these olfactory cilia. Okay? I think I've said all this. Just remember, these are dissolved chemicals that are interacting with very specific odorant binding proteins. When they leave the olfactory epithelium, they go up in about 20 bundles up through the cribriform plate. They reach the olfactory bulbs, which you guys can see really easily. Those little nubbins on the end of olfactory nerve 1, right? Cranial nerve 1, olfactory. From there, they go straight to they synapse there in the olfactory bulb, but that's it. They go all the way up, and that's very unusual. In fact, the only real system that does it that way. Olfactory discrimination, we can distinguish thousands of chemical stimuli. Usually there's about 200 of them, and you combine a bunch of them, and that gives you up to 1,000 or so different smells you can describe. I couldn't probably describe 4, but anyway. So there's considerable turnover of these receptors. Most things in the nervous system, you kind of get one set and they're good for life. They don't replace themselves very often. Also notice that the number of olfactory receptors declines with age. I think not only do your number of receptors drop, but you have less mucus up there to attach and to have the chemicals dissolve in. So your smell goes away with age. That's it. That's all we're going to talk about. Olfactory. Smell. Done. Now taste. Gustation. Gustation provides information about the foods and liquids we eat. I think it's really important for you all to realize the fact that you really can't separate the two. How many of you have ever sat down to a really good meal with a clothespin on your nose and really been able to enjoy it? Because what we taste is what we smell, and what we smell is what we taste. They're very closely put together. The taste receptors, the places that we taste, are located on our tongue, and then we have a few that are back in our pharynx and our larynx back behind. Most of the ones up on our tongue are clustered into these things called taste buds, and you will need to look at the microscope slides of the tongue and the villi and the taste buds in your on your tongue, because they're very distinguished looking, and that's really where we do all our tasting. There's 3 basic types of lingual papillae, or taste buds on your tongue. One is called filiform papillae. These don't contain any taste buds. This is like my cat. My cat has a really rough tongue. When she licks, it really feels rough. It's just there to provide friction. It's there so she can pick up those little kernels of that dry food out of the bowl, and get them up into her mouth. Same with milk and water, That's basically think of those as little scoops on their tongue that allow them to eat. So these filiform papillae, and we got some great slides of them over in the lab too. They don't contain taste buds; they're just there to help you eat. Fungiform papillae look like little funguses, and they have a few taste buds around on them. And then there's these big guys called the circumvallate papillae. They have as many as 100 taste buds on them. And if you guys would all like to reach out, pull your tongue out, and show it to your neighbor, you can actually see a row of those big taste buds in the back of your mouth, across your tongue. Kind of make a big V back there. All right, do it tonight in the shower, or when you're in the sink. In the mirror, you can see them. So here's kind of how they work. These are there's some back on your pharynx. I would just leave it at that and say that some of your taste buds are actually on the very back of your throat, back in there. We'll talk about those because they actually, your book says they're the ones that taste water. Excuse me? Water? That's what the book says. The water receptors are on the back of your pharynx. I always thought if water was clean and good, it really didn't have flavor. Right? I don't know. Anyway, So here's filiform papillae. We do have a few of the spiky ones up on the front of our tongue. A little bit rough. Not much in the way of taste buds on them. Here's the fungiform papillae that look like this. And if you just look at your tongue up close, you can see them as well. They look exactly like you see in that picture right there. They're pretty small. Then there's these great big huge ones that are more towards the back of your tongue, and they look like this. And they have up to 100 little taste buds, as illustrated here, around the edges of them. These guys are so big, you can probably notice if you eat a meal and then go look, these things will collect a little food down in them. They're pretty big. Here's the gustatory cells seen in these fungiforms. Here's the little taste buds around the edges in the big circumvallates. You just ought to look at these under the microscope. They're pretty cool. This is one thing I do not want you to learn. I think it's silly to learn which parts of your mouth test sweet, and which ones test salty, and which areas test bitter, and which ones test sour. Who cares? I mean, I've never seen anybody in a clinical situation, a doctor or a neurologist or somebody testing somebody's taste with Q-tips and different flavors. You either taste or you don't. So I think it's kind of silly for us to teach that and make people learn that, although I know some professors do. So here's these taste buds. Here's the circumvallates. Here's the row of the ones that are in the V back here on the back. One other word you ought to know about, but once again I think is kind of over the edge. Umami. What's umami? Meat. Yeah. Where do we find umami? In meat. Yeah. It's also sold in meat tenderizer. And what's that stuff that people don't like to eat that's in Chinese soy sauce? MSG, yeah. I think MSG has the umami taste as well. Is that right? You don't know? Okay. Okay, so here we go. Once again, notice out here on the surface, there's little hairs, little microvilli on the surface of these taste buds, and it's picked up. They interact with the receptors, same as the smell sort of thing. They react with the receptors, and then it sets off the electrical impulses that send the message back here. And once again, this is all part of this brain thing, you know. The only way our brain works in any way is by sending electric shocks, right? And the thing that sets off these electric shocks in the gustatory and in the olfactory system are these chemical reactions that go on between a smell or a taste and a receptor. Most of these only live about 10 days. I think that's about all I want to say. I told you that the nose ones replace themselves pretty quick. These do too. The taste, however, doesn't go straight to the brain like smell did. It does go back to the solitary nucleus of the medulla and then onto the thalamus and on up to the sensory cortex. If we decide it's a useful smell or if it's significant enough that we want to think about it. You know, we're going to assume that it's not the same as the fan and the projector. Get it? Most tastes are pretty blah, and we don't really think about them much, do we? Except that damn blue gum that she has, which she sticks it out at me all the time.
I did nothing of the sort.
Oh, baloney. So gustatory discrimination. Sweet, salty, sour, bitter. Once again, I don't think that's very important. Here's the umami, chicken broth, Parmesan cheese. Oh, that's the one, Parmesan cheese. Yeah, that's supposed to have umami flavors in it as well. Water, we taste water by these water receptors, and the only thing to know about that is that the water receptors are not on the tongue, they're on the back of your throat in the pharynx. I think I've said that. So once again, taste is a chemoreceptor. It's a chemoreceptor. It's reacting to chemicals that fall on the tongue or on the nasal in the olfactory area and cause a chemical reaction with the receptor. There's a few things that you probably did when you were in 7th grade or so, in 7th grade science. I know they do this in 112 a lot. They test whether you can taste PTC. Now, about 70% of Caucasians can taste it, and it's pretty yucky tasting, actually. And 30% don't taste it at all. That's because it's a genetic inherited taste. Okay. End of story. I don't think there's any clinical significance. I mean, people that can't taste PTC, don't have any problems with that. They just can't taste it. Number of taste buds starts to decline rapidly by age 50. Grandma and grandpa are pretty much into comfort foods later in life. They don't appreciate all the spicy, all the 5-star dinners anymore because, you know what, they just don't taste as well as they used to. Tristan, who's that? That's Pretty, yeah. Does she have another name?
Elizabeth Taylor?
That is Elizabeth Taylor, yes. And I use that slide to introduce the eyes. Now we're going to go to the next sentence, which is the eye. And I remember hearing the guy that does all the interviews, Larry King. Larry King was interviewed for the 5th anniversary of her death or something, and said, "What do you remember about Elizabeth Taylor?" He said, "Well, he had interviewed her 15 or 20 times in her life," and he said, "What do you remember about her?" He says, "I remember her eyes. She has eyes unlike anybody else. They put a hole through you. They're beautiful, and there you go." I guess. Okay, I can go for that. Cat Stevens, one of my great all-time philosophers. I always knew looking back on that my tears would bring me laughter, but I never knew that looking back on my laughter would make me cry. And you probably wonder why we talk about crying. Because part of eyes is tears. And we're going to talk about that. Hey man, how you doing? Good to see you. Okay, I'm going to show you a few clinical slides here and tell me if you think anything looks funny about them. Anything look funny?
And there's like a film over top of the whole eye.
Looks like it's kind of cloudy. Yeah. You notice this area out here, we call that the sclera, the white part of your eyeballs, right? And you notice how this is all red. See how big the blood vessels are? Looks cloudy deep inside. Anybody know what this is?
Is that macular degeneration?
No. Good guess. That's a cataract. That's right. We'll learn about cataracts. Cataracts are when the lens of your eye. The lens is what focuses the light. It's a clear thing, and when it gets cloudy later in life, you get a cataract and you can't see through it. This is something has anybody ever seen this before? A picture like this? Anybody know what this is a picture of? This is a picture of the back of your eye. And if you've ever gone out at night and flashed a light into your cat's eye or your dog's eye or your friend's eye, you notice how you get that red light refract back. Usually with cats it's green, I think. But this is a view through the ophthalmoscope. We will all look through the ophthalmoscope at the back of each other's eyes, and this is what you'll see. This is the disc in your eye. These are the big blood vessels, the arteries that come out and travel around on your retina, which is the back surface of your eyes. And I love this part of the class because most of the students have never seen this. They never knew that that was in there all the time. And really, all you do is just walk up with a light and a lens that corrects the refraction, and bingo, that's what you're looking at. You're looking right into the back of somebody's eye. You're sort of looking into their soul, the way I always thought about it. There's a whole bunch of diseases that can be diagnosed from looking at the back of the eye. I believe this is macular degeneration. This area over here is your macula. We'll get to that in a few minutes. This is another view through there. You notice that this part over here is all cloudy, and this is in focus over here. This is a retinal detachment, where the retina has detached and let go of the back of the eye, and it's just flopping around in the eye from here over. Retinal detachments can occur for lots of reasons.
So can somebody see with that at all?
Not in that part of their eye. They can't see at all. And that'll be clear to you later on. And if you don't get the retina back down on the surface, and then they kind of laser it, kind of weld it back on in place, they'll lose that vision forever. So,
Is there a time frame for that? Like,
What?
Is there a certain time frame? You need to have it done within 48 hours?
I think so. I'd say pretty quick. I don't know the exact hours. These are little bitty aneurysms, little bleeds that have occurred, where these tiny little blood vessels in the eye break and they bleed a tiny little bit. These are called microhemorrhages. This is a diabetic's eye. Little blood vessels give out. These can also be fixed with lasers. Anybody sent their kids to school with this lately? This is pink eye. Pink eye is usually caused by strep or staph. Very, very common bacteria that are found everywhere. But if you get it in your eye and it really starts growing like this, it's incredibly contagious. Take a kid, send that kid to school tomorrow, half the kids in the class will have it. Why? Kids are always putting everything in their mouth and in their eyes and so forth.
And you said it was caused by strep and what else?
Usually strep and staph. What's this? Little abscess. It's in the sty. That's right. It's in one of the glands, the sebaceous glands that keep your eyelashes oily, just like you have sebaceous glands attached to every other hair in your body. You get one in your eye, and it ends up looking like that. These are kind of fun. I didn't do eye surgery, and people didn't come to me for eye problems, but I have drained probably a dozen of these things. It's really cool. You put some numbing drops in their eye, and let them blink their eyes a few times, let their eye kind of go numb, and you just take their upper lid, and you kind of flip it over backwards, and you get right up underneath this area here. Once you flip it over, then you just take a little needle and stab into it, and the pus drains out, and it's cured. Goes away. Simple thing. This is an abscess back in one of the tear ducts. Does anybody know where your tear gland is located? Right over here in the middle?
No, up here.
Yeah, it's on the upper lateral side of your eyeball. Most people think it's over here because that's where your tears go out of your eye. But it's actually located right back up here. And this is one of the ducts that goes back there that's gotten infected and clogged off. It's called strabismus or amblyopia. Kids, we talked about the eye muscles last term, cranial nerve. Babies who are cross-eyed like this have a problem, obviously, because they're seeing double. Now, what really happens is, if they aren't treated, their brain gets tired of seeing two of everything, so it just suppresses one. And if left long enough, the kid will actually go blind in that eye, because he gets so tired of seeing double that he will lose vision in that eye. So the treatment in the early stages is to just put a patch on the other eye. So you put a patch on their good eye, the one that follows you when you move your fingers around, And then they have to use their eye muscles more on that eye and strengthen it and hopefully bring them back into line. If you can't bring them back into line, occasionally you'll have to go in here and cut the medial rectus on one side or the other. Medial rectus muscle, remember? Take a little piece of the muscle out, and that lets their eye float more out and line up.
Is that nerve disorder or is that something with the muscle?
It's a brain and nerve disorder. Yeah. Perhaps our eyes need to be washed by our tears once in a while so we can see life with a clearer view again. You know, you ladies have such a great ability to cry. It's so powerful. Right, guys? Right? When women cry, you just melt into a ball of uselessness. And why, when women cry, and you guys do this too, and crying is a good thing, why are they always doing that sniff, sniff, sniff in their nose kind of thing? What's that all about? You ever thought about that?
It's the ducts, right?
What ducts?
The internal ducts, the lacrimal ducts.
Well, what about them?
The tears are flowing through into the nasal cavity.
Right. That's where our tears normally go. We're constantly, whenever we blink our eyes, think of your eyelids as windshield washers, windshield wipers. They wiping the fluid off. It goes over here to the middle portion, and then it goes down into the back of your nose, where normally it just evaporates and you never think about it. But if you're making lots and lots of tears, it kind of overwhelms the system. You got to do this. Get rid of it. It makes your point very well. All right. So let's talk about the accessory structures of the eye. They have to provide protection, lubrication, and support for the eye. Now, what are we talking about here? Well, we're talking about the palpebrae, which are the eyelids. Remember that muscle we call the levator palpebrae superioris that lifts your upper lid? And it's innervated by what nerve? Oculomotor. Very good. The superficial epithelium of the eye, which is the actual outer surface. So if your eye is open like this, there's a very thin membrane of epithelium that's across your eye there. Call it the conjunctivitis. You've heard of that term? That's pink eye, basically, when you get an infection in there. And also the lacrimal apparatus, the tear apparatus. So, review your muscles. We're going to learn it again. I told you we were. Close your eye, open your eye. What does it take to move your eyeballs around? What does it take to focus your lens? What does it take to open and close your pupil? Okay? We'll go through these one by one here. There is a sacredness in tears. They are not the mark of weakness, but of power. They speak more eloquently than 10,000 tongues. They are messengers of overwhelming grief and unspeakable love. Whoa. Washington Irving wrote that back in the 1700s. Legend of Sleepy Hollow. Wow. Have you read the Legend of Sleepy Hollow? All right, let's learn the parts. So if this is the palpebra, this is the upper palpebrae, the upper lid, and this is the lower one down here. Notice that your eye has this big white area. White area is all the sclera. Sclera is just a really tough white fibrous layer that gives your eyeball shape and strength. And if you all want to go out here on the lateral side of your eye right now, and what we call that ballotting the eye, where you actually push on the eye, be like pushing on a football, and see how far you can push it in based on the pressure. You're going to learn that after you've felt a few eyeballs in your life, it's pretty easy to determine whether there's low pressure, normal pressure, or high pressure in the eye. Another name for high pressure in the eye would be glaucoma. Glaucoma is increased pressure in the eye. We'll come back to that. All right, so over here in the middle, we have this thing called the medial canthus. That just means the place where the upper lid and the lower lid come together. So therefore, this over here would be called the lateral canthus. Notice that the space between the two lids from here to here is called the palpebral fissure. It seems like a silly term to me, but anyway. And then most of your eye, about 70% of the surface of your eyeball is covered by sclera, this white thing. But the front part is colored, and the line between the white part and the colored part is called the limbus, the corneal limbus. The part in the very middle of that is called the pupil. The pupil is the part that's open, that's letting light come in. So if you're in a dark room, your pupils are going to probably be dilated. And if you're in a bright sunshine, they're going to be really pinpoint, really small. This over here is called the lacrimal caruncle. That's that little nubbin right in there that collects sleepy tears in the morning. When you wake up in the morning, you know you got that little bit of grit over there in the middle of your eye that you're supposed to wipe out that the Sandman left when you were sleeping. What? Eye boogers, that's right. I couldn't have said it better myself. Okay. Now, another thing to notice is that you can't normally see any white below the eye, you can't see below the corneal limbus down here, and you can't see above it. You can't see white above it. So think about that. I mean, look at your neighbors and look at me, and you'll see that, you know, your eyes close down on your eye enough so that you can't see white either below or above. And when you can see white below or above, you got a problem. We'll talk about that. All right, where's your tear gland? I've already asked that question. Now, here's another diagram that's really good, and let me just go through it for you as well. It's got a few more things on it. The last one didn't. Here's your lacrimal gland. Here's your tear gland way up here on the upper outer side of your eyeball. Notice this is your, here's the middle line, here's your nose over here, so this is the medial side. We're looking at the right eye of this drawing here. The lacrimal gland duct, see the lacrimal gland has several ducts that carry the water into the eye, just like the windshield washers on your car. You push the little button and it squirts some water out. Same sort of thing happens here. Just like the windshield washers on your car have a little bit of soap in them, this has a little bit of some chemicals in it that help you dissolve stuff away to wash your eyes off really good. I mentioned this already when I showed you the video of the girl that had the Bell's palsy, but I can't tell you how important it is that we can blink our eyes and get our eyes wet and keep them wet, because without that they dry up very fast, and they just don't, they get really sick without the tears. So the tears are constantly washing from this side of your eye over to the medial portion over here by the medial canthus. Actually, the lacrimal punctum, the lacrimal caruncle, are the two little bumps over there where the water goes to, that then goes on down here through these guys, the superior lacrimal canaliculus, the superior tear canal, and the inferior lacrimal canaliculus. They enter over here into this thing called the lacrimal sac, and then they go down through this thing called the nasolacrimal duct, and then they go and then they dump out here just below the inferior nasal concha. And that's where you get the, right, from the tears in your nose. See the inferior nasal concha there? Okay, so here's your inferior oblique muscle. Remember, inferior oblique is one that twists the eye. Here's your superior oblique muscle that twists it in the other direction, and the superior oblique is the only eye muscle that goes up through a trochlea. Remember what nerve makes the superior oblique work? Four, which is called the trochlea. Right. See, it got its name from this little pulley up here. Okay, so then the one on the very top is called the superior rectus. The one on the bottom is the inferior rectus. You've got a medial and lateral rectus, which aren't illustrated in this picture here. There's always a bunch of orbital fat in the back of your orbit, in the canal that's in your skull that holds your eyeball. It helps cushion the eye. If somebody hits you in the eye, it allows your eye to kind of retract a little bit up against a nice soft fat to protect your eye, because your eye is pretty important. Conjunctiva is the outer layer that I talked to you about that covers the eye, that covers the sclera. The conjunctiva doesn't actually come across the colored part. Lacrimal apparatus, the tear apparatus, makes the fluid, distributes it, and also removes it on the other side. The fornix is where the palpebral conjunctiva joins the ocular conjunctiva. And of all the things in this set of diagrams that they don't really, well, I think there is a picture coming up here. But basically, think about this: when the conjunctiva, which covers your eyeball, goes up, and it gets way up to about here, and then it goes over, jumps over, and gets on the inner surface of your eyelid and comes back down. That leaves kind of a little cavity up in there, which is called the fornix. So if you want to see my fornix, you see it's this area right up there underneath my upper eyelid, right up under there. Disgusting. My kids used to do that flipping. You ever do that? I think that's cool. Let's see what's cool about that. Made you think, didn't I?
Yeah.
All right, so the lacrimal tear glands, they contain the lysozyme. Lysozyme is an antibacterial enzyme. You really do want your eyes to be clean. It's nice to have a little antibiotic squirted in there to keep the bacterial count down on the surface of your eye. Clear tears, eventually, as they wash across the eye, they go into the lacrimal lake over on the medial side. Then they go through the lacrimal puncta into those superior and inferior lacrimal canaliculi. Then they go into that lacrimal sac. Then they go into the nasolacrimal duct to reach the inferior meatus of the nose, below the inferior concha. You probably wonder, why would I stress the fact that there's all those different things that it goes through? Well, yes, there is an entry in the Guinness Book of World Records. There's a record for shooting liquid from your lacrimal caruncle. So, extra credit possibility for you all sometime.
What if we can do that?
No, you don't have to do this. That's double. You can do it. All right, here's what you have to do. You have to take a glass of milk, and they use milk because it's pH balanced, right? Plus it's white, so you can see it better. See this all these little bubbles of milk going out there.
That's so filthy.
Huh?
That's so filthy.
Now, give me the route, using all your anatomic knowledge and experience, and tell me, if you were to take a swig of milk in your mouth, what route and what structures would it go through until you could squirt it out into the jar like that? Think about it. eyeball itself has 3 layers. Has anybody ever torn apart a tennis ball or a basketball? You notice how they have several layers to them? Well, it's the same thing with your eye. Your eye has 3 main layers, and you really need to know these 3 layers. You need to know them because they're quite different from each other. They have different functions, and when you understand the layers of the eyeball, it will help you understand how your eyeball actually works. Okay? So, there's an outer fibrous tunic. That's the one you can see from the outside. It's either white, it's the sclera, or it's colored, part in the middle. The middle layer, between those, just inside of that, is called the vascular tunic, also referred to as the uvea. Now, the uvea is important because it's what carries the blood to your eyeball, right? I mean, blood is pretty important, and so there you have it. On the inside, the innermost layer of the eye is your retina, or the inner neural tunic. It's the part that actually sees for you. It's neural. It's nerve cells, right, that interpret light that allow you to see. Okay? All right. The outer layer is simple. Let's just do it like this and say that this is the fibrous layer. It's actually a good bit thicker than the single line I'm drawing here, but let's just say that is the fibrous tunic, and the fibrous tunic is in 2 parts, and that is the sclera out here and the cornea out here. So it's just clear out here. And it's clear out there. Think about that. It's clear out there. Do you think it has blood vessels running through it? Probably not. It's clear. And so if you were going to die tomorrow and you wanted to donate parts of your body, would you consider donating your corneas? I would hope so. It's a pretty simple thing to do. It's the one thing that you can transplant, other than skin, that you can transplant from one human to the other without removing it while the heart's still beating. So you can die, and I remember when I was in practice, I would get the call from this crazy old guy down at the nursing home, and he would say, "Dr. Sessions, I understand you lost a patient today, Mr. Jones." And I'd say, "Yes." I said, "Well, we're going to come pick up his body. I'm from Smith and Son, a nursing home, a funeral home, and I want to know if you would mind if we took his corneas out." I said, "No, I don't mind. What does his family say?" And of course, a lot of people have talked to their families about donating their corneas. And so that's something that you can donate up to several hours later. And the reason is because there are no blood vessels in it. There is no blood flowing through it, so therefore you don't have to worry much about rejection. Okay? It's like putting a piece of plastic on somebody's eye who's had a corneal injury. All right. So the cornea is in front, white every other part. What are the functions of this outer layer, this outer fibrous layer? Basically, it's just a tough outer layer. It keeps the eyeball round because it's under some pressure from the fluids inside. It does protect your eye from getting a stub stuck or something. And it's obviously clear in front so you can see through it. Now, you also need to know that the eyeball is hollow. We're gonna dissect cow eyes here in the next day or two, and when you go into the cow eye, you're gonna find that it's just a big old thing of jelly, pretty much. That's just fluid inside. The eye is divided into two cavities, a smaller anterior cavity, and I'm going to kind of use my lousy drawing skills to put a lens in here and say that this area up here is the anterior cavity, because it's in the front of the eye, and the posterior cavity, which is this great big one back here, called the posterior cavity, also called the vitreous cavity. Okay? All right, so there's the two cavities. One of them is much bigger. It's back in the back. One is much smaller. It's between your lens and the front of your eye. Okay? Better pictures to come. Here's the way it kind of looks. If you think back to this, let's just do a little quick review here. So here's the outer layer, which is this white layer, which is the fibrous tunic, fibrous layer. And you see that it's the sclera over most of the surface, and it's the cornea, the clear part, over the front. Okay? It's divided really by the lens, and this apparatus right here divides the anterior cavity from the posterior cavity. Now notice that the thing that holds the lens in place, if this is the lens, it's held in place by these little strands right here we'll get to in a minute. And these are muscles right in here called the ciliary body. And then notice that it also extends up here in front and goes in, and this is the iris. So the iris is that part of your eye that's colored that gets big, smaller, or gets bigger. Okay, that's the iris. The part that's open here in the middle of it, you see between the two irises coming in from either side, is the pupil. Okay? And they're not really coming in from either side, they're coming in from all the way around. So this is the division line right across here between the anterior cavity and the posterior cavity. We're going to divide this anterior cavity a little bit more based on the iris. Come back to that in a minute. Here's the big posterior cavity. The third layer is the neural tunic, which is basically the orange part that you see here on the inside is the retina. Retina we're going to study in kind of detail because it's what you see with. It's where the light beams coming in interact with the retinal cells to make sparks to send the message back to your brain so you know what you're looking at. And then on the outside of that, there's a pigmented layer. And the pigmented layer is there for one reason, and really just one reason at all. And that is, if the inside of your eye were light colored, it was white, and you were in a well-lit room, and the light came into your eye, what would it do? It would bounce all around in there because it's light and bounces off the white walls, okay? And so we don't want that to happen. We want it to come into the eye, we want it to hit the retina one time, one place, and then we want it to be eliminated. We want the light then to be absorbed so it goes away and doesn't bounce around inside your eye. And that's what this pigmented layer, this pigmented part is for. Now, I've always thought it's kind of weird how the retina is on the inside of your eye here, but it is. And so light, in order to get to your retina, has to come in through the front of your eye, in through your cornea, in through your anterior cavity, through your lens, and get focused, and then land back here on the retina somewhere and get seen. Okay, yes?
I know this is a stupid question. Does light bounce better for people with lighter colored eyes? In other words, could they see better in a low light situation than brown eyes?
They might see better in a low light situation, but in bright light they see worse because it's just so bright for them because the light is bouncing around in there. But but even people with light skin generally have a pretty good pigmented layer here. Okay, now let's stop for just a minute and let's talk about the lens. We're going to come back to it, but let's talk about it. How many of you have ever had an old 35 millimeter camera with the lenses on it? Yeah, and so when you want to, you're looking through the viewfinder, you know, and you're trying to focus on something that's way out there in the parking lot, and you turn those things until it comes in focus, right? What is actually happening inside of your camera, inside of your lens piece, your nose piece? What's actually going on while you're turning this thing? That's a good question, isn't it? That's why I asked it of you.
I understand how it zooms, but
What?
I understand how it zooms. but
Okay.
focusing.
Well, basically, the whole deal is when you're turning that knob, the lens is moving forward and backward, right? And as the lens moves forward and backward, it determines where it's focusing. Back there on the back part of the camera, which is where the film is, in the old days when they actually had film. Now, the way our eye works is not like that at all, okay? We don't move our lens forward and backwards when we want to focus on something far away or close up, okay? Let me throw one more thought at you to let you think about this. Remember the good old days when you did wood burning with a magnifying glass out in the sun, and you would take the sun, and it would come down, and you'd take that lens in your hand, and you'd work it up and down until you could get that light to focus into a little bitty point, right? And then it would burn the wood. Maybe it's how you started your neighbor's house on fire. Did you?
No, she says also knows ant burning.
Ant burning, yes. There you go. Gosh, I never even thought about that.
My husband used to do it to G.I. Joe's.
Oh. Well, anyway, the point being that when you're moving the lens like this, you're doing the same thing as you are in that 6mm camera. But that's not the way we do it in our eye. Anybody know how we do it in our eye? We change the shape of our lens. That's right. So we change our lens from being really thick, where it brings the light in a lot, to making the lens get really thin, where it doesn't refract it so much. Okay? And the way we change the shape of our lens is by these little fibers here that stretch the lens out. So we can either stretch it out to make it thinner, or we can let it go in to let it get thicker. All right. Now, you remember that story I told you last time about the front seat and the back seat?
Yeah.
All that stuff. You haven't forgotten it yet, have you? Motor and sensor, right? All right. Well, here's a way that will help you remember this. When I was a kid, I liked to read, and we would go on all these long trips in the car, okay? And I'd be reading and reading and reading, and my eyes would just get so tired and get sore from reading. And I would say to my mother, "Mom, my eyes are just tired. What can I do?" And what would she say?
Stop reading.
And no, she can't say that.
Make the light brighter.
No.
Close them. Suck it up.
You guys
Drink some water.
are so good. Drink some water, yeah. Smoke some dope. That's what they do for glaucoma these days. No, she would say, "Look at the mountains. Look out in the distance." And so I would look out in the distance for a few minutes, and sure enough, my eyes would feel better. Why is that?
Oh, because you're not focusing.
Because
so
you're not having to squeeze these muscles down, and actually these muscles are pulling out, okay? They're working to make your lens get thicker so you can read up close, okay? So, the way our eyes work is not by changing the focal length, not by moving the lens, but rather by changing the focal length of the lens itself by making it either thicker or thinner. Okay? And so the muscles are relaxing when you're looking in the distance, and the muscles are working hard when you're trying to read something up close.
So can you strengthen them just as any other muscle?
Sure. Read in the car more. Yeah.
Eyesight that way.
What?
Could you correct eyesight that way?
No. We'll talk about eyesight in a little bit. But that's a good question. All right. Now, there's so many things about the body that I just find so incredibly incredible that it just kind of blows me away. Think about this one for a second. I'm going to stand over here, and I'm going to look at that periodic table of the elements over there on the far wall. You can all do this too. And then I'm going to look at this thing right here, right in front of my eyes. Okay? I'm going to move my eyes from here to there, from here to there, from here to there. And in the 10th of a second it takes me to go from there to there, my eyes focused. So when I look at that, it's in perfect focus, and when I look at that, it's in perfect focus. All the time. Our eyes can focus faster than any automatic single-lens reflex camera in the world by about a factor of 1,000. That's how good our eyes are, how quick they can change, these fibers can change in order to change the shape of that lens in order to keep it in focus for us. That's pretty cool, isn't it? Gina, don't you think? Yeah. Okay. All right, let's keep working our way through this. Here's the, so here's that little fornix thing I was telling you about that's up underneath your eyelid, right there. You see, because here's the ocular conjunctiva, and the ocular conjunctiva lays on the front of the eye, and then it goes up in there, and then it becomes the palpebral conjunctiva, and it covers the inside of the eyelid up there. And it leads to this little lake back in there, the fornix. There's one up there, there's one down here, where your tears can collect. But they don't too much, because usually you just blink your eyes a couple times and all that water works its way over to the medial punctum thing and goes on down into your nose. All right. Julia, what's the name of that muscle right there? That is your, yes.
Superior rectum.
Ah!
Elevator palpebral.
Ah!
I thought I got it wrong. I think that's what
Orbicular.
That's the orbicularis oris. Remember, it's the one that goes around your eye. And so when you slice it like that, you're cutting it crosswise. That's what it does go around. Right. The levator would be back up in here, way back up in here, and then it would go this way.
They're in your eyelids?
Yeah, that's how you close your eyelids. Go.
Yeah.
Your ocular forest.
Ocular. Oris is
It goes
mouth.
around and around. Basically, I'm just asking you to look at the direction the fibers are going here, and see they're going this way. You're cutting them on, cutting them sagittally. Do something to think about. What's this thing right here?
That's the conjunctiva.
No, the blue area, the blue thick area.
Oh.
The fibrous tunic?
No, that's in the eyeball here.
Okay. What?
That's the cartilage that you have in your upper eyelid. That's what enables you to do this crazy
Don't do it.
flipping your eye thing up there, because you're flipping that cartilage up. That's the cartilage in your upper eyelid. So you have one down here in your lower lid, but it's so much shorter that you can't really do it very well. All right. Okay, a couple more things to point out. The blood supply to the eyeball itself, for the inside of the eyeball comes in through the back of the eye, along with the optic nerve. In fact, this blood vessel here, called the ophthalmic artery, imagine that, comes right in along with the nerve right into the back of the eye, into that disc that we looked at on those pictures from the ophthalmoscope, and then they branch out and go across, run across the eye like that. And then the veins come back and bring the blood back out. Okay? We'll talk about that more later. All right. Now, you might think it would be neat to have your retina go all the way around the inside of your eye. But what would be the use of that? Because the only part that you can interpret is the light that comes in from the front and goes out here onto the retina here, right? Light that hits the retina out here is something you could see. You can't really see in this area up here because the light would have to bend or something. So you don't have retina up here at the front, above halfway up. And the line around your eye from the inside, which is the end of the retina, is called the ora serrata. Now the reason it's called the ora serrata, serrata means
Serrated.
close, what?
Serrated? Broken?
Serrated. Anybody got some of those, mother had scissors that have the serrated edges, it's back and forth, back and forth, back and forth. Well, when we dissect our eyes, you're going to see the edge of this, which is where basically where the retina ends and where the non-functional part of the eye really begins, and it's called the ora serrata because it's serrated along the edge. Okay. Don't get worn out on these pictures because these pictures are so cool. Can you believe there's an entire medical specialty devoted just to this 1 cubic inch of the human body? These eye doctors are amazing. They have to know so little. Okay, so here's another picture, and once again, now you can see the colored part of the eye up here, which we call the iris, right? The iris, the part that gets smaller and gets bigger. It's the part that makes you have blue eyes and you have brown eyes and so forth. Okay, it's the colored part of your eye. That's the iris right here. Okay, and notice that the iris has muscles in it. We're going to learn about them in a minute, but it has one bunch of muscles that are circular, and when they contract, it gets smaller. And then there's another group that are radial, they're going outwards, and when they contract, it pulls the edges back to open the eye up. Okay. Now, which set gets innervated by the sympathetic system, and which set gets innervated by the parasympathetic system?
The set that gets the radial would be sympathetic.
I'll just let you think on that. Okay. So I think we've been through most of these parts. Here's that ora serrata that we just mentioned. This thing here, right here, is called the ciliary body. The ciliary body is kind of the culmination of where these muscles here insert onto these little ligaments here, which are called the suspensory ligaments, as I recall. Suspensory ligaments, yes, of the lens. And those are the ligaments that pull the lens out to make it get flatter or let it come back in to get it and let it get thicker. Here's the medial rectus muscle out here. Here's the lateral rectus muscle over here. And this is really stupid, but you see this right here? See that little vein coming into the eyeball? I don't think there is any such thing. I think that is there to keep other book manufacturers from copying their picture. Because if that thing shows up on some other book manufacturer's diagram of the eye, they're going to know they stole it from them. Because it's not, it shouldn't be there, I don't believe. It doesn't make sense, yeah. Map makers do the same thing.
In regards to the iris, color of the iris, what evolutionary purpose does color or function serve? Because there isn't any light coming through it.
Correct. Why we have brown eyes and blue eyes.
Everybody used to have brown eyes. There was like an evolution of eye.
I don't know the answer to that. I would perceive that, I think blue eyes are pretty. It might change who you reproduce with. Just a thought.
I cannot remember what I was watching, but there was a thing about eye color, and it's like everybody used to have brown eyes, and then
Yeah.
it was like an evolutionary change.
I've never seen a blue-eyed monkey or chimp. If you think about
Why?
it. Yeah, I
Okay,
don't
cool,
know.
like puppies.
Here's one other point I want to make to you here off this picture, and that is the fact that this is the disc over here, and you notice that it's off center. It's off to the side, and the disc where it comes out of the back of the eye does not have retina there, so you can't see from that area. So it's actually a blind spot. Okay, your disc is a blind spot. We'll do a test over in lab, and you'll see how we all have a blind spot. It's kind of right up there. We've got a little thing to teach that to you, show you that. But this is a really important thing. This is called the fovea. The fovea, another name for the fovea is the macula. You guys have heard the term macular degeneration. The fovea is where you do about 95% of all your seeing. Okay?
How much?
About 95% of it, I would say. Maybe I'm just guessing at that number, but think about it. If I want to look at this can of Red Bull, am I going to look out this way and use the side of my retina to look at it? I'm going to turn my head, I'm going to move my eyes, I'm going to put it right on the word that I'm reading, right? The point is, we have such good eye muscles and so forth that we always look with the part that's straight out front, and then coming back in the back of your eye, it's right here, it's called the macula. That's where our finest vision is, and that's the one we use all the time because it is the best vision. It has more cones and more rods. Actually, it has more cones, but it does have rods too. We'll talk about those. Okay, I think I'm going to stop right here after this big fancy picture and tell you that we're going to go to lab, we're going to go over the test, and then we will, and just know that you would be well served to take a few minutes between now and the next lab to read about the eye dissection, the eyeball dissection, and then a lot of the eye stuff we learn off of models. You know, we have some really good models over there. Questions? Make sense? Yeah?
Is the lens black?
No, the lens is clear.
So
It's
what
totally
makes our
clear.
pupil black?
What?
What makes our pupil black?
It's just that when I look into your pupil, I'm looking straight back to your retina. There's nothing there to reflect the light to me. So it looks black, but it's basically clear.
So is that why I need flash photography level to be pushing? Because it's reflecting the
Right. And if you get the flash out to the side, then that's like getting the flash out over here, and it comes in here and flashes off the disc, and you can sometimes see a white spot. Okay? Yeah. All right, we'll meet over there in a few minutes. Okay, here we go. This is the second lecture of special senses. So we're starting into the eye. We're well into the eye, I should say. Okay, so we talked about the eye the other day, and we talked about kind of the stuff that's in this picture. There's several things I think it's really important that you understand, that there's 3 basic layers to the eyeball itself. The outer layer is the fibrous layer, which is the sclera, all the white part, and the cornea up front. So that's the outer layer. It's just pretty much tough stuff there to give you an eyeball. The second layer, next inside of that is the choroid layer. The choroid layer involves the blood vessels that supply the eye, supply the retina and so forth. But up front, the choroid becomes a little more variable, and it is differentiated into the suspensory ligaments here, the ciliary body, which is here and there. Ciliary body has muscles in it that control the shape of the lens. And then also the choroid separates up here into the iris. The iris is the thin layer that sticks up into the anterior chamber that is really the pupil, which is the open spot, and the iris, which is the part of your eye that's colored. And you know your iris, in addition to being colored, it has muscles in it that allow your pupils to either open or close, depending on the amount of light. Okay? So I think we're going to take another run through this as we advance on. The vascular tunic, this is going back to the parts of the second layer, which I was just mentioning to you, actually. The vascular tunic, also called the uvea, it provides a route for the blood vessels, as I said before, and so it supplies the tissues of the eye. We call that the choroid layer that has the blood vessels and lymphatics running into it. Up front, there is the iris, and the iris is the thing that opens and closes, gives you blue eyes or brown eyes, opens and closes to let in air, let in light rather. And the choroid up front secretes and reabsorbs the aqueous humor that fills up the eye, the front part of the eye especially, in the ciliary body. So just going back to that last picture, the ciliary body here, in addition to having these muscles in it that stretch or unstretch and change the shape of the lens, this also makes aqueous humor. Aqueous humor is the name given to the fluid that's inside the eye, especially up front, the front parts of the eye, the clear fluid that you see through. Okay? Now the rest of the eye is filled with fluid also. We'll get to that in a few minutes. Now not only does the choroid body make the fluid, but it also reabsorbs the fluid to take it out. Okay? And we'll go through that step by step here in a few minutes. So that's the ciliary, going on in the ciliary body, and it controls the shape of the lens, which I mentioned is essential to focusing. And I think I've said all I need to say about that. Let's talk about the pupillary muscles. The pupil is part of the uvea. It's part of the vascular tunic. And I just think it's important that you guys keep a kind of loose outline in your mind as to what's in the fibrous tunic, what's in the vascular tunic, and what's in the neural tunic, which is the inside, the innermost layer, the retina itself. Okay? So the pupillary muscles are made out of part of the choroid layer, the middle layer, the uvea, or the vascular tunic. And notice that the muscles are shaped like this. So there's one set of muscles that are the pupillary constrictor muscles, and they are a sphincter. And just like every other sphincter that we have, sphincters are circular. When they contract down, they close off, and so the pupil would get smaller when the constrictors contract. Does that make sense? There's also these radial fibers. See the fibers that go in with the radius of the eyeball going in this direction. And when they contract, they pull it out and make the eye bigger. Make the pupil bigger to let more light in. Okay? So the constrictors constrict, the circular muscles constrict it, the radial muscles pull it open. Okay? And you can see dilated here, little pupil here. You can do that to each other over in the lab. We'll have little lights for you to check each other's lights. The ciliary body is this area up front, out at the sides. It extends posteriorly to the level of the ora serrata. The ora serrata, I mentioned it the other day, it's that serrated edge that kind of marks the end of the retina coming from the back and the ciliary body up front. You can't see with any of the inner lining of the eyeball anterior to the ora serrata. So several things are here in the ciliary body, the ciliary processes and the ciliary muscle. Ciliary muscle attaches to the suspensory ligament of the lens. Now remember I told you the story about me riding in the car reading the book?
Mm-hmm.
And I said the way my mom told me to make my eyes feel better and relax was to
Look out.
look out. And when you look out, what does that do to the shape of your lens?
It flattens it.
It flattens it, that's right. So, therefore, we need to use our muscles to make our lens get real thick, right? So which direction do the muscles of the ciliary body go? They are circular, right? They're sphincters. So when they contract, when they get tight, that's what makes the pupil, the lens get thick. Wrong picture. Makes the lens up here get thick. So keep that in mind as well, okay? It always seemed funny, you think, oh, well, the muscles are really working when they're pulling the lens out. No, they're really relaxing when the lens is out. They have to pull together to pull it in, to make it get fat. Got that? Does that make sense? Think it through logically. Think it through with what I said about looking far away and relaxing. And once you get it, it'll make sense forever. Yes?
It also helps to think about when we're studying muscles. If you use a muscle, it's contracting. So
Right, muscles can only contract.
when they're in use, yeah. So
Yeah.
That's what.
There you go. All right. The choroid layer is what brings the oxygen and nutrients in. Remember, they're bringing in the blood vessels from the back of the eye. And now the third layer we'll talk about for a minute, and that's the retinal, the neural tunic, which is the retina. The retina is the part of your eye that actually sees. Reminds me of medical school. We were making rounds one day. We were on this course called Physical Diagnosis. It was one of the very first clinical courses you ever took, and I was with this group of maybe 4 or 5 other medical students. We were going around from bed to bed. We're in these big wards with 25 patients in a room, and I remember the old doctor said to me, he said, "Well, Dr. Sessions," they always called us doctor even though we weren't. He said, "Would you please check this man's eyes?" And I said, "Yes, sir." And so I pulled out my little light and I shined it in his pupils, and I told him to watch my light, and I watched his pupils get bigger and smaller, and I looked in there and I had him close his eyes and open them and so forth. And finally the doctor looks at me and he says, "Okay, Dr. Sessions, now, what are eyes for?" I said, "Well, they're to see with." He says, "Have you checked that?" I said, "No." He said, "Well, just remember that, because you can examine eyes till the cows come home, but if you don't ask the patient if they can see, what have you really accomplished here?" And, so I felt embarrassed and stupid, but you know what? I have never forgotten it to this day. The first thing you got to do is be able to see if they can see. Make sense? All right. So the retina is the part that has all the nerve cells in it, and it contains the visual receptors. And the receptors, the things that the cells that are in our body that allow us to see, are called rods, and they're called cones. Okay. Now, the way the book tells you to remember that is rods are what you use to see when you're seeing in the dark. Cones are the more specific kinds, and they allow us to see color and clarity. Now, think about this. You get up in the middle of the night, it's really dark in the room. Maybe there's a little bit of moonlight coming in through the window, but that's the only light at all. And yet, you can probably open your eyes and still see the furniture in the bedroom, see well enough to negotiate across the bedroom and get to the bathroom. That's what we all get up for in the middle of the night for, right? All right. Now, when you're seeing that, think about the last time you were doing that in the middle of the night. Are you seeing in living color?
No.
No. You're just barely seeing shapes. It's totally black and white. There is no color to it. And that's because you're only seeing with your rods. Now, the thing about rods is they can see with just an absolutely infinitesimally small amount of light. Just the slightest little bit of light in the room, and you can see. You don't see clearly. You don't see in color. You don't see sharp things. You wouldn't want to try to read something. But the fact is the matter, you can see. And that's because you have rods. They are highly sensitive to light. It only takes a tiny bit of light for you to be able to see with them. Cones, on the other hand, are the ones we use, that we're all using right now to see. They're the ones that give us really good clarity. They are clustered. You have a whole lot more of these guys right around your fovea. Remember the fovea? Fovea is that part that's directly through your eye. It's the part that we all do all of our looking with. I'm going to have to come read that, you know. You know that.
They're about your shoes.
You like the shoes?
I just said they're girly color. That's super embarrassing.
These are my new shoes. They are Portland Airport rug colored, and they're from Keen. A great shoe company, by the way. If you've not bought any Keens, I have several pairs of them, and I bought these because I saw them down there. And they're the most comfortable pair of clogs I've ever worn, Miss Garside, and I wore them today just for this point.
Oh my god.
I wanted you to realize how fun color can be. And you see color with cones. Writing notes in class. My goodness. All right. So the cones are really centered right here, back here around the fovea, which is also called the macula. Okay, it's the main part of your eyeball that you see with. And because we have these great things called eye muscles, and we can move our head, we don't really have to look with the outside parts of our retina. Now, you know, if I'm sitting here looking at you guys, and I put my hand out here and I wiggle my fingers, even if my eyeball's looking straight at you, I can see my hand out there. Do I see it very clearly? No, but I can see my fingers moving. And so I know that that part of my retina is working, but the part that I always look with, and you all do too, is with our macula. So we just move our eyes and look at what we really want to look at.
Is that considered the yellow spot?
The what?
The yellow spot?
No. The yellow spot is the disc. It's over here. The disc is actually a blind spot. I'll go through that in a minute. Yeah.
But the fovea is in the center of the macula.
Correct.
The fovea centralis.
I think, yeah, fovea centralis and macula mean the same thing. Does that make sense?
That's what we-
Is that the way you got it?
Yeah.
Okay. Yeah. Okay. Now, this is kind of a close-up, obviously, of the back of the eye around the disc. So here's the disc right here, the optic disc. and you notice that the disc is where all the blood vessels come in, and then they fan out from the disc to go out through the retina, supply blood to the whole inner lining of the eyeball. But outside of that are all of these cells right here. These are the rods and the cones, okay? Now, we're going to look at the retina under the microscope, and you need to look at it and understand it, because the way this works is here's rods and cones on the outside, on the inside of the eye. The light is going to come in from the front of the eye up here. It's going to go in through the lens, and it's going to come back here, and where it hits on the retina is what we're going to see, okay? Now, once that retina stimulates these rods and cones right along here, it takes that information and it sends it up here to some of these other sets of cells that kind of put the picture together, and then the nerves run this way and they all run out through the what? What's this big nerve called? The optic nerve. And where does it go from the back of the eye? To the optic chiasm. And which fibers cross over?
The medial.
The ones from the medial part of the retina cross over. The ones from the lateral sides of the retina don't cross over and go on back to the nuclei where they synapse. Please notice, this seems weird to me. This always seems backwards to me. If you've got the light up here coming into your eye, notice that that light has to flow in through these nerves, these tiny little nerve fibers right here, before it gets into these guys. But that's the way it works. Then it passes the information back forward, and then through these axons right here, it goes out and back to the brain. Now, do those little nerve fibers get in the way? Well, sure they do. Are those little things in focus? No, they're not in focus, so you don't really see them. You don't see them in your vision. Plus, they've been there your whole life, and you just kind of suppress them. That make sense? All right, we'll go through that a little more in detail in a bit. Now, here's looking at in the eye, and when you look in the eye, you see the disc right here. Here's where the blood vessels both come out and go back in. Remember that all the nerves are going to feed over here to the disc. And because of that, there is no retina here, so it's a blind spot. Okay? We'll prove that to you over in the lab. This is the part that you're actually doing your best seeing through. Call it the macula, call it the fovea, either one. Think macular degeneration. When people get older and their macula degenerates, their vision degenerates, because that's really that's the tiny little part of your retina that you actually have always done all your real seeing with. Okay? All right. There's several layers of cells here that I mentioned to you here, but I really think you need to learn these in the lab. The neural tunic, the retina, consists of several layers of cells, one being called the bipolar cells. The rods and the cones synapse on these guys with ganglion cells, and we don't want to embarrass her, do we? The horizontal cells, they all group together, and the amacrine cells are comparable to the horizontal cell layer. Once again, this is where these bipolar cells are synapsing with the ganglion cells. Now, that's a bunch of malarkey, what I just said. But here's the way I want you to think about it. When I'm looking at this screen up here, from where I'm standing, this doesn't look like the letter S to me. What it looks like is a bunch of dots that are black, and then a bunch of dots that are white. Okay? And if you were all standing up here, you'd see the same thing. You can probably see it too, can't you? But the picture is created by a bunch of dots. Now, if you go home tonight, and I'm not sure this works so well on these new fancy flat screen plasma TVs, but on the old kind of TVs, which is really what this is here, it all worked like this. For example, let me go to this. And if you look right up in here, and you notice that looks like a blood vessel running through there. But if you get up in here close, you'll see that that's really not red. It's really a combination of several dots of different color. And, you know, how many of you guys have wired video systems together? And you know there's these wires that are called VGA, right? And there's red ones and there's yellow ones and white ones. I'm thinking the three main colors are red, white, yellow. No, that's the wire colors, but the primary colors that we really see with red, green, blue. Think RGB. That's the word I'm looking for. RGB cables. Red, green, blue. That's how you get color TV. These colors right in here are not created by. It's mostly red, but there's also a little green and there's also a little blue to give you the different colors that are in there. Like right here, you know, there's white and blue and green. All three make up that blue line. There's lots of dots of different colors. Well, think about that when you think about your own eyes, because we don't have single cells back there that can pick up all three colors so much. And the way we create a picture is by a bunch of retinal cells, billions of retinal cells back in there. They're all seeing a dot. And then the brain, through these horizontal cells and bipolar cells and amacrine cells, are processing those colors together to put them together so that your shirt looks like it's green and yours looks like it's gray and my shoes look like they're the color of the Portland airport rug. But they're very colorful. All right. So you're going to be able to identify these cells under the microscope pretty easily. You just got to get the picture, look at them, and what they're doing, what all of these cells are doing, is processing, forming an initial process of the picture before it goes to the brain. The brain then has to take those images that come in and say, oh yeah, that's a human face that I'm looking at.
That's amazing.
It is amazing, isn't it? It's incredible, this stuff is so amazing. Okay.
So it's all of the cells that do that?
What?
It's a combination of all of the cells.
These cells that I've just mentioned, yes. So these guys are going to facilitate or inhibit communication between the photoreceptors and the ganglion cells. It's going to suppress some of them, and it's going to add sensitivity. By suppressing some and augmenting others, it's going to increase the sensitivity of the retina. The optic disc is that thing we just talked about. It's the blind spot. It's the origin of the optic nerve. It's where the optic nerve goes out of the back of your eyeball. Nice picture here in a schematic form. These are the pictures you'll see in lab. We're looking at it from this side, so this is just like this. And by the way, I'll tell you, of all the things that I have to go up to students that they're in lab at a time like this, and they're looking like hell through their microscope, and they've got their picture laying there, and I have to go up and say, no, wait a minute, it goes like this. Because you got to be real careful to be sure you've got the inside of the eyeball, which is where the blood vessels are, the inside of the retina is on the inside, and these guys here are on the outside. Please notice that this layer is the one you can always tell when you're looking under the microscope. You can always see this pigmented part. The pigmented layer looks like this. It's really dark, and that's to cancel out the color. After the light comes in through here, it gets recepted by these different rods and cones, which are these guys right here, and then the messages start sent back out through these amacrine cells and through these bipolar cells and through these ganglion cells. The message comes back out this way, and after being processed by these things we just talked about, it takes a turn and flows through this nerve layer over to the disc and then goes out the back of the eye. Okay? But the retina, this pigmented layer is so the light doesn't just keep bouncing around inside your eye. It goes through one time and that's it. It gets absorbed. See this little arrow down here? Most important light label on the whole slide. That's the way the light comes in. Let's talk about the chambers of the eye real quickly. I think they're pretty easy. We said to start with, there is a large posterior chamber. That's the big chamber in back. And then there's the smaller anterior chamber up front. The smaller anterior cavity. Be careful with your words here. Large posterior cavity, also called the vitreous chamber or the vitreous cavity, and a smaller anterior cavity, which is up in front. The anterior cavity extends from the cornea up front to the iris. The posterior chamber goes from the iris back to the ciliary body and the lens. So both this anterior chamber and this posterior chamber are both in the anterior cavity. So they're both up front. Okay, so be careful about your words there. Aqueous humor is this fluid that circulates within your eye. It diffuses when it's circulated around. It goes out through this thing called the canal of Schlemm. I just love that word, Schlemm. Anyway, then it re-enters the circulation in the bloodstream. Intraocular pressure is what we need. We need intraocular pressure. We don't want our deflate gate. We don't want our footballs to collapse, right? We have to have a certain amount of pressure in them to keep our eyeball expanded and keep it round. That's a good thing, but we don't want too much pressure in there because then that leads to problems. That problem is glaucoma. Okay, so I wrote this little slide and stuck it in here for you. Don't forget the difference between cavities and chambers. There's two cavities, a little anterior cavity and a big posterior cavity. The chambers are both in the anterior cavity. Okay? So this might be another way to look at that same thing. This is the posterior cavity back here in the vitreous cavity, back here in the back of the eye. It goes all the way from there all the way back to the back. And up front here, from the lens forward, is the anterior cavity, as illustrated right here. And it's divided into the anterior chamber and the posterior chamber. The posterior chamber is just this area right here, behind the iris, but in front of the lens and the suspensory ligaments here. Now, remember how we followed the route of the circulation of the cerebrospinal fluid? Let's talk about the route of the circulation of the aqueous humor, fluid in the eyeball. It's created right here in the ciliary process, in this little ciliary area right here, and it goes out here into the posterior chamber, both in just right in there, as it's illustrated here. And then it comes out through the posterior chamber and goes across here, and then here's the pupil, the part you see through, and the pupil is open, and the fluid just comes out through here and comes up here into the anterior chamber. It circulates around in the anterior chamber. That's where you get little floaters. You guys have all seen little floaters floating by inside your eye from time to time. They're in the anterior chamber. And then when the fluid is ready to leave, it goes out through here, goes right over here to this little canal, which is right at basically at the edge of the iris, and enters the canal of Schlemm. The canal of Schlemm goes back to blood vessels, and it goes back into the bloodstream. Okay? Now, if the canal of Schlemm is clogged, you get a buildup of fluid, you get glaucoma. If it's making too much fluid, you can get glaucoma as well. How do we treat glaucoma? Anybody? Yeah?
You can put a bleb in.
A bleb?
Okay, you're gonna laugh at me. I saw it in the documentary.
Okay.
It's like a little hole, and then they leave like part of it open so that it can drain.
Okay. Where does it drain? Just outside?
It kind of looked like a blister, didn't it?
Yeah.
The blood was like a blister.
Blister, but it's like up here.
I'm not familiar with that. We just
Going
used,
blind.
we just used to always give them some eye drops.
Oh, that was one of the other things. They
Yeah.
gave eye drops.
And the eye drops make them not make as much fluid. Yeah. And the way you check the intraocular pressure is a lot different than it used to be. In the old days, you actually had to numb their eyeball up, and you go out here to the side, and you lay this thing on their eyeball. It's like a disc that has a little thing in the middle, and then the little thing in the middle is pushed down, and as it goes down, it made a little scale, a little wave thing go up and show you exactly what the pressure was. Now they just come up to you with a little thing and it blows a little air into your eye. Is that whiskey or something good?
No, it's water. You want some?
The way you were sneaking it, I just wasn't sure. Here, my water.
That is real rough.
And then it just blows a little puff of air on your eye. And that little puff of air causes your eye to distort a little bit, and they actually can measure the pressure just from that. It's kind of an ultrasound thing. Okay, let's talk for just a minute about the vitreous body. It's the big thing in back. In humans, it's not really water in there. It's more like Jell-O. It's gelatinous. It doesn't change over much. It kind of stays, you get what you got, and you pretty much keep it your whole life, just about. It's just there to stabilize the shape and to hold the retina down against the eyeball. Let's talk about the lens for a minute. The lens is really cool. The lens is made up of this material called crystallins. Crystallins is a protein that is clear, but yet it can change shape. That's pretty amazing if you think about it. It's clear, but yet it can change shape. So there's no nuclei, there's no organelles in these fibers. They just sit there in the lens. It's the only place in our body that we find them. And a cataract just means that the lens has lost its transparency. Now, there's several diseases that can lead to you getting cataracts, one of which is taking steroids. But most of people just get it from being old, and their lenses gradually cloud over. You can actually see it from the outside if you just could shine a light at somebody's eyes. Usually, if they have a real, what we call a mature cataract, you can see it pretty easily. Now, let me see. I'm going to talk about that in a minute. Let me just tell you my little story about cataracts, because I used to take a team of surgical people, surgical nurses, surgical technicians, and usually other doctors down with me, and a few nurses. When we go down to Mexico, there was a town called, I want to say Zihuatanejo, but that's the end of Shawshank Redemption. Not there. I'll think of it in a minute. Down about 1,000 miles south of Mexico City. We'd fly in there, and there was Northwest Medical Teams ran a clinic down there. They had built a clinic and a school. It was really a great thing. What they would do is they would bring the smartest kid from a bunch of towns all around. They'd bring in about 30 kids a year, and they would just go to the town and get the elders of the town to tell them who they thought the smartest kid in the town was. And they would accept them into the program, and the kids would come down to this clinic, and they would go to school for a year. But the people in the mountains of Mexico down there, they're not what we would all think of as Mexican folks. They basically are from Indian tribes, Zapotecs and several other groups down there. And they're really short. They're like this tall, max, adults. And because of the racial discrimination, I think you could call it, in Mexico, the Mexican government has never invested any money in that part of Mexico. There's no roads, there's no schools, there's no nothing. Most towns don't have any water at all. So they take these kids, Northwest Medical Teams was taking these kids, bringing them in, and teaching them for a year. But you know, in addition to teaching them, they also fed them for a year. And some of these kids would grow 8 to 9 inches in a year because they'd never had really good food. It was just amazing to meet these kids and work with them. But one time I went down there, I took Jay Hoffman with me. He was an ophthalmologist from out in the Dalles, and I said, "Come on, let's go down there for a couple of weeks, and you can do cataracts, and I'll do, general surgery." He said, "Great, let's do it." So I'll show you pictures later. I think I have pictures of it. But it was so amazing because a cataract is such a simple thing in this country. My parents got to the point where they couldn't read sessions in great big letters. They couldn't read the headlines in the newspaper, and they had their cataract operated on when they could still read little print, But people down there couldn't read anything. They were totally blind. So these old folks would walk 30, 40, 50 miles to come to the clinic on days when they knew there was going to be an ophthalmologist there, and wait and wait and wait. And Jay would operate on as many as he could. He would do 7 or 8 a day. He would only operate on one eye because he could do twice as many people if he just did one eye on each patient. And that's what he did. And then the next morning, they would sleep around the clinic. There was no beds or anything for them to sleep on. They would just sleep on the grass usually with their families. And the next morning, he would have them come back in, and him and his nurse would take the patch off their eye. And it was unbelievable because these people could now see. And then some of them hadn't seen for 20 or 30 years from either eye. Take the cataract out, and they can see perfect. It was really incredible. So I had my eyes open to this cataract business. Now, down in Mexico, he actually took their whole lens out, and you could see it. It was about the size of, about that big around. It was all brownish looking, and it was that thick. You couldn't see anything through it. In America today, they don't even take the cataract out. They just go in with a little phaco emulsifier. They kind of stick it in through a needle hole on the side of the eye, stick it in there, and it throws out an ultrasound wave, and it just dissolves all the cataract. It all falls apart, and then they just suck it out. And then they put a new lens in, a plastic lens that fits in the middle of their eye. It's incredible. So it took, if we'd had all the equipment that he uses to do cataracts up here, he could have done 50 a day. As it was, he could do 5 or 6 a day because they actually had to make an incision in their eye, go in there and take the whole lens out and so forth. But that's what cataracts are all about. It's a big deal. It makes me think in America we take them out too early. I mean, come on. Just because you have a little bit of trouble reading the finest of fine print, you really need to have surgery for that yet? Come on, why don't you go blind first? Got to do something to deserve an operation like that, All right. This is a picture of Josie, my granddaughter. She's bigger than that now. And here's the question for you. Why do swimmers, she was a racer in those days, why do they wear goggles? For the chlorine. Well, yeah, okay, chlorine. Any other reason? To see. Oh, wait a minute, you can see when you just jump in the water, can't you?
It's to help with their refraction, right?
To help with their refraction. Ooh, she's been here before. Probably.
No, I just read the book.
You read the book. Damn it, that's great. Yeah, because our eyes, we've spent our whole life living in air, okay? So our eyes and our brain is accustomed to the level of refraction that is provided by air being in contact with our cornea in front, okay? But when we jump in the water, we have a different level of refraction. We now have, instead of air up here, we have water. And water has a different coefficient of refraction than air does. Now, what does that mean? Well, look at that. And there's no lens involved there, is there? But why is the spoon broken? It's because for us to see down here, the light has to travel through from the spoon to the water to the glass to the air. Then it comes to air and goes into our eye. Up here, it goes from the spoon to air to glass to air and then into our eye. Okay? And because of the fact that the level of refraction is different between air up here and water down here, it looks broken. Okay? And for those of you who have never gone swimming with goggles on, believe me, when you've got goggles on and you've got air in front of your eyeball instead of water touching your eyeball, you can see perfectly clearly. Right? You can see where the lane lines are, you can see where the ropes are, you can see where the wall is when you're swimming fast and running up into the wall. So all of that stuff. So that's really why people wear goggles, is so that they have air in front of their eyeball instead of water in front of their eyeball. So it fixes that refraction problem. Well, let's talk about that. Light refraction is the bending of light by the cornea and the lens. Now the cornea is the clear part across the front of your eye, right? It's part of the fibrous tunic. And it's going to refract the light as the light comes in from the air, goes into the cornea, goes through the cornea, goes out from the cornea into the anterior chamber, which is water, again, refracts it again. The reason that we as humans don't see that well when we're in water is for that reason. And the main source of refraction in the human eye occurs at the air to cornea interface. That's where the main refraction occurs. So that's why if we can just keep air in front of our eye, we're probably going to be okay. And these other things aren't going to matter as much. All right, a couple other points you need to understand. One's called focal point. A focal point is the specific point that the light intersects on the retina. Now, what does that mean? Well, remember that business with the lens and the sun, and you're trying to burn your initials into a piece of wood, and you've got to work that lens up and down until you get the light beam focused on one little point. And when you get it there, you hold it, and it heats up, and it burns the wood. Okay? Well, that's focal point. And the focal point is different for every lens, every fixed lens, like that glass lens that we've just been talking about. Okay? So that's why when you are in the sun over there, you move it forward and backwards until you get it to the right focal length, the right focal point. And indeed, the focal distance is a term that means what's the distance between the center of the lens and the focal point. Now, when we're trying to see, when we're trying to get our brain, get our retina to see things, where do we want the focal point to be of that light that came in through our lens, goes back on our retina? Where do we want it to be? We want it to be on the fovea, but we want it to be right at the level of the retina. We don't want it to focus in front of the retina, and we don't want it to focus behind the retina. We want it to focus right on the retina. That's what I was trying to drive. All right. A couple other words you ought to know. Accommodation. What does accommodation mean? Well, our lens accommodates to look at different things at different lengths. And so when I look at the periodic table over there, and then I look at my finger and want to focus on my finger, my lens accommodates for that. And you can go back and forth. It's kind of cool because when I'm focused on the periodic table over there, and my finger is here, even if I put it right up here in the middle, it's totally out of focus. Y'all ever check that out? And that's because I haven't accommodated to my finger. Now, if I move my eyes so I'm looking right at my finger, my lens will automatically accommodate so it focuses right on my finger. It gets thicker, right, to focus on my finger. We're going to show you I'll show you some examples of that in a minute. What's astigmatism? Anybody got astigmatism in here? You know what that means? So what do you mean by that?
Well, it makes it so that you can't see things really sharp and clear.
Yeah.
They're always kind of blurry.
They're always kind of blurry. Anybody got a better explanation than that?
Well, I was saying it's not perfectly shaped.
Right. Yeah. You know, you'd like to think that your lens is nice and square like this. I mean, nice and straight. So that if it gets thicker, then it focuses, and if it gets thinner, it focuses on it at a different level. But astigmatism means you've got like a bend in your lens. And that can go this way, it can go that way, it can go that way. You've just got a deformity of your lens so that it isn't a perfect shape. And so no matter how much you accommodate it, you make that crooked lens thicker or thinner, it still doesn't make a perfect picture because it's bent. That's what astigmatism is. This is your lens. You see I'm bending it. Get it? Now, I have an astigmatism, and so technology is so amazing because I go to the eye doctor, and he's doing all these tests on me, and then he figures out I've got an astigmatism. He says, okay, now you tell me when it's the sharpest. And he takes this special lens that he's got in front of me, and he starts to turn it. And he turns it, and then you say, oh yeah, that's it. It's perfect. Well, that's him identifying which direction my lens is bent, basically. And then he can correct that by putting the opposite bend into the lens by grinding it a little different. So it's not ground perfectly round. It's actually got a little divot to it to correct the astigmatism that is in my lens. Isn't that amazing? Visual acuity. What is visual acuity? How good you can see. Let's just say it's how good can you see. And what is normal? 20/20. What does that mean, 20/20? You can see perfectly from 20 feet away?
Level of detail seen at a distance of 20 feet.
Okay. All right, so let's say we define that level of detail as these words right here, Portland Community College. Okay? Now, I'm 20 feet away, and with my glasses on, I can read Portland Community College. Okay? So that tells me, if I do it with one eye, I'm 20/20 in this eye. I have normal vision because I'm 20 feet away, and we've decided that if you can see that size lettering from 20 feet away, that's normal vision. Okay? So what does it mean if you're legally blind? And what would be the numbers for somebody who's legally blind? Not legally blonde, legally blind. Oh, come on, don't read that damn book. All right, well, somebody's legally blind in this country, I believe, when you're 20 over 100. 20 over 200, it depends from state to state, but I'll buy that. 2200. Now, what does that mean? 2200. So that means somebody at 200 can stand 200 feet away if they're blind and they can still see it?
No.
Okay, what does it mean?
Well, the person that has 20/20 can see the same thing at 200 feet.
Exactly. That's right. So what 2200 means is I can stand with my normal vision 200 feet away, and I can still read something that somebody that's 2200 has to be up there at 20 feet to read. Okay, so if you're legally blind and you're 20 feet away, you might be able to read trash over there on that big sign because you're blind, pretty much. And so you would be able to read it here as a blind person, but a normal person
So shoes.
would be way back here and can read it from 200 feet. Seems backwards, kind of, doesn't it? Okay? So what does it mean if you're 2012? No, that's backwards.
They have to be
Right. If you're 2012 and you stood right here at 20 feet and you could read it, let's go down to a smaller level of print than I can read, because you got better vision than me. I would have to be able to go up here to 12 feet in order to see what you can read from 20 feet. Okay? I want you all to go home and think about that tonight. And once you think about it and get it down, you won't ever forget it. But just remember that 20/20 is normal vision. Higher numbers are lousy vision, up to 2200, which is legally blind. I don't think you can drive in Oregon above 2080. Is that right?
I think it's even 2050, 2100.
Yeah.
I mean, that's pretty. You wouldn't be able to see any of that.
Yeah, but that's with corrective lenses.
Well, that's true. You can get a driver's license, which is with correction only. And believe me, I don't go out without my glasses, even though my eyes are still pretty darn good. But when you're less than 20, it means when you're 12 over 20, your vision is better than normal. Okay? Does that make sense?
Yeah.
Y'all got that now? You'll never forget it, right? So that's visual acuity. Acuity means the accurateness of your vision, I guess. Let's talk about this. This is the business of accommodation. This is changing the shape of your lens in order to focus on different things. So here is something, you're trying to focus on this red dot. Okay, so this would be like me trying to focus on this pin right here. Okay? To focus on that pin and to get the light to be in focus on the back of my retina, I would need to have my lens be very thick, like this, in order to bend those rays in tightly in order to get them to focus right here on my retina. That makes sense? If I'm looking at something long off, like I'm sitting here looking at the periodic table, I don't need it to bend that sharply. Here it comes. My lens is now flattened out. See how it's flattened out in this picture compared to that one? And yet it still focuses on the back of my lens, so I can see perfect. Something that's a long distance away. That's normal vision, okay? Now what happens if your lenses are screwed up a little bit? What would be the difference in those two pictures if you were nearsighted or farsighted? I never can keep those two things straight, by the way.
Well, nearsighted people can see things that are close to them, but they can't see far away.
Okay, nearsighted people can see things up close that are near. Okay? So what would be the difference in those lines? I just wanted you to think about that for a second before I showed it to you. So this is normal. I don't care if you learn this word here. This would be myopia. Now, in this case, you see the lens is refracting, but it's not refracting it enough because it's not focusing on the retina here. It's focusing it in front of the retina. And so this person, this retina, these now are going to cross and go out, and that's going to be very blurry looking to them because the lens is not refracting it appropriately. On the other hand, hyperopia, which is the other side of the coin, is now it's not refracting it enough, and so instead of focusing it up here, which would be out of focus, and it's not focusing it right on the retina where it needs to be, it's focusing it behind the retina. That person's not seeing well either. So both of those people need corrective lenses because their lens is not working just right. And it's working, and it's still accommodating. It's changing from force, but it's off a little bit. And so, the first person that was focusing right here, at this point, now we put a diverging lens in. You notice it's concave on this side, and so it diverts the light out. And so it comes into your lens, even though your lens is a little off. Now, with that lens up front, it focuses just right on your retina here. That makes sense? Now, if we went to that other case, instead of putting a converging lens that's concave up front, we put in a lens that's shaped like this, which is convex. Okay? And the convex lens is going to bring these lights in a little bit, then this is going to bring them back out a little bit, and it's going to focus them right there. And so that's the difference in the shape of the lens that people get if they are either nearsighted or farsighted. One of them is going to get a concave lens, and one of them is going to get a convex lens. Now, I can promise you that the problem that we all have is that we're so damn close to normal, all of us in this room, I can just bet you, that you can hardly look at these lenses and see a difference. They're so close to being flat, but the grinders that the ophthalmologists use are so specific and so perfect that it's just a tiny bit of difference. You know, these diagrams like this, showing it really bulging out on both sides, it ain't that way. Yeah.
So you have bifocals.
I do. I have bifocals.
So how, that's for seeing near and far, right?
Yeah.
Focals are
So
for
what is
seeing.
that? What's your issue?
Okay. My problem is really the other one, I believe. I don't have any problem looking at a distance. My eye can accommodate perfectly. The lenses above, they don't either magnify or subtract. It's just plain glass from here up. But when I want to read something up close, I'm really blurry, and so I have stuff in the bottom of them. When I'm looking at things at a distance, I really don't need glasses. And I shouldn't say that because the last set of glasses I got actually have like a half a diopter or something, so I can see it changes it just a little bit on the upper half. But that's what bifocals are all about. A lot of people don't need much on the top when they look at things away from them, but up close they do. Does that make sense? Presbyopia is a kind of hyperopia that occurs in the elderly, like me.
So that's farsighted.
So, I guess so. I can see okay at a distance, but I can't see up close. So you tell me whether that's nearsighted or farsighted. Because I really do. I have a hard time knowing what those words mean. Did I steal this from somebody? No. All right, let me talk to you about LASIK for a minute. This is a really interesting thing. What we're talking about here is lenses, right? And whether you wear lenses, glasses, or whether you have the lens inside your eye is working right. But if the lens inside your eye is off a little bit, like it is in me, people wanted to try to figure out, well, how can we change the shape of the eye so that people don't have to wear glasses? We can just operate on the eye and change the shape of the eye so that it'll correct everything. Now, there was a lot of debate about that. And the first debate was, do you consider it a disease to wear glasses? Tristan, do you consider it a disease to wear glasses?
Yes.
You do? Okay, then your insurance ought to pay for it, right?
Yeah, big deal.
And they do. Well, most people, mine does. Because it's Medicare. And God hope you can all get on Medicare someday. Anyway. Well, that set the laser guys to seeing, well, how can we change the shape of the lens, change the shape of the cornea, the thing that's out front, right? The clear part in front. How can we change the shape of that? Because if you could buckle it out or buckle it in a little bit, you could correct by changing the shape of your eye. You could change the refraction and change the accommodation and everything, so you wouldn't need glasses. And that would be really great for people that, you know, want to skydive or they want to do underwater stuff or whatever. I mean, a lot of people just don't like wearing glasses. And they think they don't look good in glasses. Me, I don't even care. But I think
You can get contacts.
I look great in glasses.
You look fantastic.
Thank you. All right, well, here's the deal. So they came along, and as these newer and newer lasers were coming out, let's say this is the front of your eye. This is the cornea in front. Here's your iris back in here, you know. Here's your lens back in here. Okay? They thought, if we can just change the shape of this thing out here, we can compensate and do away with glasses. And so these guys said, well, we've got this laser that we use to cut things. You've seen those lasers that cut into metal, and, you know. We've got one that's really gentle and doesn't cut very deep, and we think we can start cutting into eyeballs with lasers. And that's really what LASIK means, laser-assisted I don't know, or radial keratotomy, basically. So what they decided to do, so here's the cornea out here. And let's look at it from the front now. We're looking at it from the front. Here's the cornea. They took a laser and they started making these radial cuts. They made a bunch of them, like that. They basically cut part way down through the cornea. So maybe they're cutting in this deep. And what that then allowed the cornea to do was, because of the pressure in the eye, boom, it bulged out a little bit. And when it bulged out a little bit, that corrected the shape of their eye so they didn't have to wear glasses. I remember sitting in the doctor's lounge when guys were starting to get these things. They were all going up to Canada to get them done because they were a lot cheaper up there. And my question was, well, guys, you know, you're getting surgery for something that is it really a disease? You know, and they're cutting your eye, because the thought was, well, what happens if you're cutting the eye like that, and the laser cuts a little too deep? Or what happens if 10 years from now, that eye, which is now bulging out like this, starts bulging out more? Now it's bulging out to here, and now it's bulging out to here, and then one day you wake up in the morning and there's a blob of eye out on your bed where the front of the eye just ruptured. Well, that hasn't happened. You'd be glad to know. They've come up with new lasers that are even more controllable. They can control the depth, and they put a lot more slices than they used to make it even more even. The biggest problem with this is that they can't sometimes avoid people who get their distance vision fixed with LASIK. They need peepers to read up close. So they're having to wear reading glasses now. But there's some new system that I saw advertised on TV the other day that they've now tried to fix that too. it's pretty amazing if you think about what they're doing, and people are signing up for it. So I shouldn't complain about it. Yeah.
One thing that I have heard, because my dad has looked into it, there's like, well, and my uncle did it, but he did it, and then they fixed it, and this was like 10, 15 years ago, but he needs it redone,
Yeah.
and they said they couldn't do it because there wasn't enough cornea. So like, if you have a thicker cornea, I guess they can do it a couple
Kind
times.
of creepy, and the
Yeah, it's really.
whole thing is kind of creepy. I've heard that too.
And then my dad said what you're talking about, where they like they fix one eye for farsightedness and one eye for nearsightedness.
Oh, that's great.
So.
How you doing? Oh,
Well,
wait a minute.
that's
How
kind of,
you doing?
well, that's basically
Oh, how
what
you doing?
they basically explained it to him. I'm sure there's, other things involved, but that's basically how they explained it to him.
All right. Moving on along. Let's talk about a few last things here, I think. Rods, I said before, those are the ones that you see in the dark with. They respond to very, very little light. Almost any photon will fire off your rods. Doesn't matter how much energy content it does. It doesn't do colors. Cones are much more sensitive. They have many ranges of sensitivity. They can see from very bright to down to reasonably dark, and they can also see in colors. I don't expect you to learn the chemistry of rods and cones. I think that's just a little bit too much for this course. But I do want you to know this. I want you to know that there's one chemical that you should know, and that is called rhodopsin. And rhodopsin is opsin plus retinol. Retinol is important because retinol is the chemical that is synthesized from vitamin A. And remember, your mother always told you to eat your carrots because they have vitamin A in them. It'll help your vision. Well, here it is, right here in A&P, so you can go back and tell your mother, thank you for making me eat my carrots, because the doctor in A&P said, yes, it does help your vision. So here it is, retinol from vitamin A. It reacts with this rhodopsin to make you see. Now, I'm going to run through this, but please don't take notes on this. I just don't think it's very important. This is how we see chemically and how it works in the rods and cones. A photon of light comes into your eye, and there is this chemical here. Here's the rhodopsin, which is the cis-retinol and the opsin. Remember cis and trans? Some things have the molecule hooked on that side, some have them hooked on that side, and they're kind of isomers of each other. Well, it takes and turns this cis, where the thing is going this way, and the photon of light changes it to being like this, which is now the trans-retinol. The process of changing the shape of that molecule, is what opens up these membrane pumps and allows sodium, that really leaks, and lets the sodium leak in, and that's going to fire off a nerve action potential, right? That's going to go up to your brain. Okay? Actually, it's going to go into one of those other cells. They're going to process it. It's eventually going to get to your brain. But it is a matter of cyclic AMP changing shape. It's a matter of the rhodopsin changing shape that we talked about over here, and closing the channels, which then stops the reception, and it changes the voltage along the rod, okay, or the cone. And it's once you change that voltage, then boom, it sets off an action potential, which sends it up to your eye. Up to your brain eventually. That's about all I want you to know. I just want you to know that you need rhodopsin, you need retinol, and that it's the light, the photon of light changes the shape of the chemical, which is what causes the message to be sent to your brain. Does that make any sense?
Say that one more time.
Shut up, Sessions. Just
Photon of light.
photon of light is cool. How many of you remember Roy G. Biv? My 7th grader knows Roy G. Biv. Red, orange, yellow, Roy G, blue, I is indigo, and violet. the spectrum of light that we see and based on wavelength. And basically, because light comes in many wavelengths, we have different kinds of cones that interpret different colors, and that's how we see in color. So you can see we've got red cones, we've got green cones, and we've got blue cones. We also have rods, but you see they're just in black and white. Red dress, blue dress debacle. There you go. Oh yeah. Oh, yeah, I forgot about that. I never did vote on that one. Color vision and color blindness are something you need to know about. We have some wonderful color blindness books over next door in the lab that every one of you should flip through and see if you are colorblind, because I bet most of you have never been tested for color blindness, and you may find that you're blue-green colorblind or whatever. It's kind of cool, but just seeing how the books work is cool too. So we have them over in lab. Color blindness is simply the inability to detect certain colors. Some people are born without any blue cones, for example. Some are born without any green cones, and that's why they're colorblind. Where does the message go? Well, you know, it goes through both the bipolar cells and the ganglion cells, and then it heads back to the brain. And enough said. Our visual area is in our occipital region, right, of our brain. Here's a standard color vision test. Can everybody see the 47 there?
Yep.
What do you see? 12. Very good. Now, this is what has to do with that crossing over stuff that I've told you about before. The left half of both visual is up here. So this retina and that retina, which you're seeing that side of the visual field, are both going to end up going to the right occipital lobe, and the right halves are going to arrive at the left occipital lobe, and I'll let you figure that one out. But it all has to do with that, what do you call that thing when they cross over? Yeah, the optic chiasm. So here's a nice diagram that shows this to you as well. And this is better in your book because these colors are hard for me to see, and maybe they are for you too. But once again, the medial stuff from the medial side of the retina is what crosses over. And so therefore, this stuff that you're looking at out there crosses over here. This stuff from here stays on this side. So this side over here sees everything out that side. And this side of your brain sees everything out that side. Okay? You notice that the synapse is once here in the lateral geniculate ganglion on its way back. What's depth perception? How far away is something? How do we know how far away it is? This is so wild to me. The reason we know this is close is because for me to look at my finger right here, both of my eyes are pulling in to look at it. Because they're in, my brain knows that that's close. If I look and it's way out there, I'm not having to pull my eyes in, and therefore I know it's farther away. It's basically comparing the relative positions of objects between the left eye and the right eye. Somebody who's blind in one eye, do they have any depth perception? No. How do they know how far things are away from them? Well, if they used to see, and now they just see in one eye, they know how big the car looks, so they figure, well, it must be about 50 feet from me because it looks about that big. They only can measure size. They can't really focus in on it. This is a uveitis. This is an infection in the fluid in the eye. I think I showed you these pictures at the beginning of this talk when you didn't know anything about the eye, really. It's really red. You look out, see how hazy it is in the eye. That's a bad problem. That person's needs to be on lots of antibiotics. Probably needs an operation to drain their eye to get that pus out of there, so it doesn't destroy their eye.
And it's an infection.
Excuse me, it's glaucoma. Oops. It's under really high pressure. It looks just as bad. I'm sorry, I forgot. This one is a cataract. I'm not going to speak up anymore. Okay, this is the disc. These are the blood vessels. Where's the macula? It's out here. That's macular degeneration. This is the lifted up retina over here. It is bled from the vascular layer, the uvea. It's bled, it's torn, and the retina is lifted up off of the retina, off of the wall of the eyeball. And this piece of retina out here is not seeing anything. So this person is really pretty blind over in there, and that needs to be operated on, and fixed, and tacked back down, or they will lose that vision completely.
When I was reading about it, it said that when they weld it with a laser, it can create blind spots.
Welding it, yes, yeah. Because it may not it's got to get flushed down with no blood under it. You know, if there's blood under it, then it can't work. Pink eye, acute bacterial conjunctivitis. This is the one I thought was the uveitis. Notice pus layering out here in the anterior chamber. It's kind of yucky looking, isn't it? That's a real bad problem.
How does that happen?
Oftentimes it's caused by a stab wound to the eye, but it can occur vascularly too. Bacteria circulate around in your blood that kind of drift out in your eye. This is a sty. It's just a little pus pocket in one of the sebaceous glands that feeds one of the eyelids there. This is called a chalazion. It's an obstruction of one of the tear ducts coming from the tear gland up in here. It gets infected. That also usually just needs to be lanced and drained, and it'll clear up just fine. These are little hemorrhages. This is a diabetic eye, diabetic ophthalmopathy with microbleeds here. They can treat this if they stay on. Was that our favorite?
I don't know if it was her or not, but I winced a little bit when I seen it. I don't think it was.
Anyway, these can be fixed with lasers. You can actually, with a laser, look through the eye and focus the laser on the back wall, and zzz, and it burns it.
Can't that cause diabetic retinopathy?
Yeah. But you can slow it down a lot with laser treatments. Here's the kid with strabismus. Also leads to amblyopia, which is a loss of vision in the one eye that it doesn't get lined back up right. Far vision, is that right? Farsightedness? Yeah. Okay. Nearsightedness? Show the vision video. All right, I'll show the vision video either in lab or next time. All right, let's go to lab.
Let's do it.
Yes, sir.
Question regarding the vascular tunic, middle tunic,
The vascular tunic. Fire away.
uvea,
Uvea, yeah.
or the choroid, which are these all interchangeable terms?
Yes. Any other questions?
Oh, we can take the quiz now, right?
No, we're still missing Susie
Oh.
and Montchill, actually.
Do you know if they're coming for sure?
Did they call me every morning? I don't know.
I don't appreciate your sass.
You don't appreciate my sass. A lot of people don't appreciate my sass. And I appreciate the little amount you have taken. All right, anyway. Actually, let me go. Well, I guess they're not going to come up. What? Yeah. That? Is that good?
Yes.
You want another one?
Yes.
Okay. Okay, this is the second or third lecture, I think maybe the third lecture on the special senses. And we're going to do the ear like I said. So here we go. Let's talk about the ear from the outside. The outside of the ear is called the auricle. The auricle refers to the big Barack Obama or Dumbo ear part out here on the outside. You see it so well made out in cartoons. I think it hears really well, actually. Anyway, so the auricle is this piece of tissue out here that surrounds your external acoustic meatus. What do you think it does? Or Simona, what does it do? Why do we have an auricle? To tunnel the noises in. That's sort of true. Protect it from being just an opening. Keep the bugs out. Oh, that's up there? Yeah. Well, the reason it's there really is for directional hearing. So, like if Miss Garside, stand up. Just stand up. And close your eyes. Don't watch me. Can you point to where the ear clicking is? Close your eyes. Where's it coming from? And just one more thing. Please don't open your eyes yet. You little cheater. And how can you, where is it now? Point. Yeah. Now, how did you know that? Think about, all of you, think about how it is that we can figure out where noises are coming from. Okay, Miss Garside, how did you figure it out? Just practice, right?
Yeah.
Okay, well, if it comes from one side of you, is it fair to say that you're probably going to hear it in that ear louder than in the other ear? Right? And if I'm looking at you, and my ears, since they tend to point this way, I would probably hear it better if the noise was coming from in front of me than if it was coming from behind me, right? And there would also be a time delay. In other words, if I'm standing here like this and you said something, I would hear it first in this ear and then in this ear, right? Not much difference, but a little bit. And your brain gets to figuring that out through your lifetime. How fast does sound travel? The speed of light? No. The speed of sound. What is the speed of sound? It's called Mach 1. And what is it? 750 miles an hour. So if you're in a jet plane, and none of you have ever been in a jet plane that's gone 750 miles an hour, I can promise you, unless you've done fighter jets, you've never broken the sound barrier, okay? And in fact, we wouldn't appreciate it if Phil Knight out here at Hillsborough Airport took off and broke the sound barrier. Why?
Sonic boom.
Sonic boom, that's right, a sonic boom, boom, would come through here every time they broke the sound barrier, okay? So, usually, even in the military, the guys don't ever break the sound barrier unless they're out at sea, away from people, because it's just too disturbing to everybody. Too much noise. But anyway, that's the reason we have auricles, and that's why they're kind of pointed forward to help us differentiate and discriminate where the sound is coming from. 750 miles an hour. It's an important thing to know. Okay, so the external acoustic meatus is the thing you stick your Q-tip in. It's the opening that goes down to your tympanic membrane, your eardrum. And indeed, if you stick a cotton swab in your ear, you'll know it when you hit your eardrum. It doesn't feel great. That's called your tympanic membrane. What is the tympanic membrane? Well, it's this real thin, and it's so thin you can see through it really easily. And if you guys have never seen a tympanic membrane, you're going to get to see one today. You're going to get to see one of your buddies in lab because we have some little ear looker things for everybody to look into ears. You're wondering what I'm wandering around up here for. This is it, as usual. Okay. In our external ear, we have those ceruminous glands. I'm sure I've told you this story before. If you stick your finger in your ear and you smell it, it smells really lousy. And that's because that wax that's made in your ears is called cerumen, C-E-R-U-M-E-N. There it is right there, cerumen. And it, you know, you can get a whole lot of it built up in your ear where you can hardly hear, and the doctor maybe even has to kind of dig it out if he's trying to look at your eardrum. They do that with kids a lot. I never did. And when I had kids that came in to see me, if I couldn't see their eardrum, I just said, wax in ear. And I didn't go digging it out to look, but some people do. But this wax, not only does it keep bacteria down, fights microorganisms, but it also smells bad so that bugs don't go crawling in your ear to take up roost. Because even bugs don't like the smell of it. I've had people, when I worked at ER, about once a month, I'd have somebody come in and say, I think I've got a bug in my ear. And I would always think, yeah, right. And it never was. It was always some wax or something in there. But I did have this one guy came in, and it was a bug. It's like a beetle kind of thing. It crawled down in there. I pulled him out in pieces. Okay, so just on the other side of the tympanic membrane is the middle ear cavity, and you guys have all heard, oh, he's got a middle ear infection. The middle ear is where your kids always get ear infections, and you may have gotten ear infections when you were a kid. And the way that I always remember the middle ear is remember that the middle ear has a hose, a tube, that connects from your middle ear up here to the back of your throat. Remember? We call that the eustachian tube. We studied that somewhere along the line, didn't we? Maybe not yet. Anyway, that's the more fancy name, the more acceptable name these days, the more politically correct name is the auditory tube. And that's why when you go up in an airplane and you start coming back down, all the kids on the plane start screaming. And why do they start screaming? Why does the pressure build up in their ear? They have bigger heads and smaller tubes. Okay, you were really close. Okay, so we adults, what do when we start coming down in the airplane? We pop our ears. We take it, squeeze our nose, and go, and you can feel that little bubble of air get shot into your middle of your ear from your throat here. Puts a bubble of air. It equalizes the pressure on the two sides of the membrane. The membrane doesn't get stretched all out one way. Comes back to normal, and it doesn't hurt anymore. Okay. And how many of you have ever been scuba diving? Yeah, scuba diving is pretty cool. And I remember diving down in the Bahamas a few times, and you would, when you were going down to, say you were going down to 60 feet to kind of get started, and you'd hold onto the anchor rope of the boat and just walk your way down really slowly. But man, I had to clear my ears about every 2 feet going down because the pressure would build up in my ear, and I'd have to equalize it by doing that with your mask and blowing air in through your auditory tube to equalize it. Inside the middle ear, there's three little ossicles, three little bones called the malleus. Malleus means hammer. Incus means anvil. Anvil is one of those things that the blacksmiths used to make horseshoes on. And if you've ever watched the Roadrunner commercials, they must have dropped thousands of anvils an hour, it seemed like, on some bad guy. And then the third one is called the stapes, which is like a stirrup. It's just rounded and has a flat part at the bottom. We'll see those. Okay, so here's the stuff I've been talking about, just so you know. Here's the auricle out here on the outside, directs the sound. The sound comes here into your external acoustic meatus. This is the one you stick the Q-tips into, and if you stick them far enough, you'll touch your tympanic membrane, which is this thin little clear piece of tissue that's stretched across here. And then it's the center of the tympanic membrane is attached to the first of the three ossicles, 1, 2, 3. And the idea of these ossicles is actually to magnify the movement of your eardrum. Now, in a real snippet here, the way we hear is because sound waves wah, 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 come into your ear, and they make your tympanic membrane go wah, 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 and vibrate back and forth. The tympanic membrane then makes 1, 2, 3 ossicles go wah, 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 and it actually magnifies the amount of movement. That's why the ossicles are laying where they are. And then the last one, called the stapes here, lives here on this little window, which is also flexible, and it's called the oval window right there. And that little wah, 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 wah gets the fluid in this thing going, and the fluid vibrates through this thing called the cochlea, which is called the cochlea because it looks like a conch shell, seashell. I can hear the ocean, yes. Okay, so it sends those little waves of sound up through here, and then they come back down the other half of it, actually. And if they're still around, they go back out here through the round window, which then deadens them, and that's the end of it. Okay, now remember that all of this, everything you're looking at there, you will never see in A&P other than in models, because you see this is all bone around it. And all of this apparatus is sunk down in the temporal bone back there. Okay? So you just don't see any of this. Notice that there is a nerve that comes in here. It's called nerve number 8, remember? The vestibulocochlear nerve. We used to call it the acoustic nerve. And it has 2 parts. One is the cochlear part that goes to this thing here that hears for you. And the other part is right here, and it's called the vestibular branch of the vestibulocochlear nerve. And it goes over here to this apparatus, and this involves the semicircular canals and the vestibule. And we're going to get to them after we do hearing. But this end of things is what lets you get dizzy, and lets you know which way is up and which way is down, and lets you feel acceleration when you stop, and makes you feel acceleration when you take off. Get it? Okay, we'll come back to that mainly. All right, so here is this middle ear cavity, and remember that the middle ear is just full of air. There's nothing in there but air. And you see that it connects through this auditory or eustachian tube out here to the back of your nasopharynx, which is in the back of your nose, right at the top of your pharynx, back in here. Okay? So if this is the external ear out here, and you get an infection in it, we call it external otitis. And I bet every one of you has had a case of external otitis. You may have heard it called swimmer's ear. That's the most common cause of external otitis out here. And you usually just treat that with some drops in your ear. And those are not ophthalmic drops. Those are called, what's another word for ear? It starts with an O. Odic drops, remember? I've got a bottle of them in my bathroom, and it says, for odic use only. And I'm thinking, why didn't you just put for ear use only, you know? So The non-A&P professors out there would know what that means. I know a lot of them don't. All right. So yeah, and so that's how you treat external otitis out there. If your eustachian tube here gets clogged up, or if it's just too short, and that's the problem in kids, little babies especially. Theirs is very short, and it's also very small, and the slightest bit of swelling from a little bit of a sore throat, or for a little virus or a little cold or anything, will make this tube here swell up. And when it swells up, then this thing, then mucus starts forming in here, and it finally gets infected, and then it's full of pus, and that's what's called an ear infection. It's called otitis media. Otitis media. So this is external otitis out here. An infection in this area is called otitis media. And there's only two ways to treat otitis media. One is with antibiotics, which is probably the way you all have taught your kids. The other way, if it's really full of pus and it's really tight in here, the doctor will go in and actually make a little cut in the tympanic membrane and just let the pus drain out. The hole in the tympanic membrane will seal back in 4 or 5 days. But that lets all the pus out and lets it clean up, and then it heals over.
So when they put ear tubes in for someone that gets chronic, where do they do where do ear tubes go?
Yeah, that's a very good question. I was just going to say, let's look at, they go right there in the tympanic membrane. So here's the tympanic membrane, and you're looking at it from the outside, and here it is. And they just go down here and they make a little incision about like that. Now, if we were looking at it from the side, so here's the side of the membrane, and they made that little cut down here, then they put tubes in that look like this. They balloon out on this side, and they balloon out on this side, and they have a hole through the middle. Okay, so these two bulges keep it in place so it doesn't fall out. And it's still got an open place to drain, so those kids can, if they start to get another infection, it just drains out through there, and it's not a problem to them. Most infections our body can handle really well as long as it can open it to the outside. Okay? Now, you've heard of inner ear problems. Granny has inner ear problems, and she has dizziness, and she feels terrible, and she's got vertigo and all that stuff. Well, that's an infection over here into this apparatus, and most of the time it's not really an infection. We really don't know a lot of what causes problems in the inner ear. We have a bunch of medicines we treat for it, but in all honesty, if you have a lot of medicines and none of them work very well, it's probably because you just really don't know what causes it or how to fix it. And that's kind of the way it is with inner ear problems. They've probably gotten smarter since I retired, but I don't know. Maybe not. Okay. Here's a couple more points. There's a couple of muscles in here that you should be aware of. One is called, is this one right here. It's called the tensor tympani muscle. Well, think about it. Tensor tympani. If it tightens up, it puts tension on the drum. Okay, so it can't move back and forth so much. And it's there to protect your eardrum from really loud noises. Okay, so that's what your tensor tympani does here, which is the same thing that this other little muscle called the stapedius. The stapedius muscle you see attaches right here to the stapes and does the same thing. Keeps it from vibrating too much, so as you don't blow out your ear, basically. Blow out the inner part of your ear over here, where the cochlea is and so forth. Okay? Remember that this whole area in otitis media, basic standard childhood ear infections, basically turns to pus. This whole thing is full of pus, and when you look through from the outside and you look through the tympanic membrane, instead of seeing it being nice and clear, you can just see the pus behind it. And also, the tympanic membrane, instead of being shaped like this, is usually bulging out at you. And that's why the doctor looks in your ear, especially in your kids' ears, to diagnose things like that. Okay? This is what this is a really cool picture. And the reason it's such a cool picture is because this picture is taken from inside the middle ear. Okay? So they got a camera here inside the middle ear, and you're looking out through the tympanic membrane. Here's the tympanic membrane. And that light that you see is somebody with an otoscope looking in from the outside. So they've gotten a camera inside the ear, inside the middle ear, looking out. That's a neat picture. And in this picture here, you can see the stapedius muscle. You can see the tendon of the tensor tympani. I'm not sure I see it all that well, but I think it's right up in there. There you have it. Okay. So, the way this whole thing works, like I just told you, is that it converts sound waves. Wah, 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 wah. Has anybody ever been at a rock concert and stood up near the front and been up near one of the big speakers, and you look at the speaker and it's going, you know, the big round parts going back and forth, back and forth. Well, that's because those are bass notes, and our ear can hear down to about 16 vibrations per second. Now, it's not exactly one, but it's not too far from it. If you've had a really good stereo system and turned the bass up really loud and listen to the low numbers, it's tiny, tiny. And then the really high notes are something like 50,000 to 70,000 vibrations per second. Now, we can't hear quite that high. Our tympanic membrane won't vibrate that fast, but it'll vibrate about 50,000, I believe. So we can hear really high notes. We can hear really low notes, and when they're that low, you can almost, it's not like you can count them, but it almost feels like you can feel it in your bones. So, remember we talked about the taste buds and the olfaction being basically a chemical reaction where these chemicals are floating around in the air and they land on our mucosa and they meet up with little receptors, and that's what fires off the signal that says, oh, this is a rose I'm smelling, right? Well, those are chemical reactions. These are totally mechanical. This is totally mechanical energy. In other words, it's going back and forth. There's a little hair cell in there. I'm going to simplify it. And that little hair cell is in your cochlea, and it's floating in the breeze. And when the tympanic membrane goes back and forth, the little hair cell goes back and forth, and that sends messages to your brain. And depending on the frequency, tells you whether that's a low note or a high note you just heard. Okay? All right. So the whole idea of the tympanic membrane vibrating is that it's going to convert these sound waves into mechanical movements. Things are going to actually move back and forth. And the tympanic membrane is going to move the three ear ossicles. The ear ossicles are going to move the stapes, which is going to move the fluid on the other side of the the oval window. And that's going to send the fluid moving back and forth in the cochlea. And that's how we're going to hear. It's mechanical. It's kind of weird, isn't that amazing? The tensor tympani, I mentioned, stiffens the tympanic membrane. The stapedius muscle does the same thing. It just attaches to the stapes, so it keeps the stapes from vibrating the fluid in the oval window too much, which could potentially damage the inner part of the ear, called the cochlea. The inner ear, let's get to it now. It's full of fluid again for the first time in this system. It's called endolymph, and the endolymph is in a little sac that is surrounded by the membranous labyrinth, and the membranous labyrinth fits in the bony labyrinth. So basically there's a hole in the bone that's shaped like that cochlea deal, and the inner lining of it is so this is the bony labyrinth, this is bone out here, and And then there's a membrane in here called the membranous labyrinth. And then inside of that is the endolymph. We have some really nice models of the ear over in the lab, and you definitely ought to study those. They will show up on the practical, but they're so cool because they really show you how this works. And I think you can all understand it pretty easy if you just take a few minutes at it. Okay, so this bony labyrinth surrounds and protects this membranous labyrinth. There are several parts to the membranous labyrinth. One is the semicircular canals, and the one is the vestibule. That's the gravity thing and the acceleration thing. And then the one we're going to talk about first is the cochlea. That's the one that's shaped like the ear, the seashell. Looks like this. So, this is where the stapes is going to attach right along here, and that stapes is going to set that fluid moving in the same frequency as the membrane is moving. Low frequencies, really low notes. Really high frequencies are really high notes. Okay. So once you get this fluid moving over in here, then it works its way up through the blue there, comes up to the top, and if it's still going, it turns around and comes back down to the bottom. Boom, 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 boom. Notice the outside is bone. The inside is all covered with this membranous labyrinth, this membrane, and then inside of that is fluid. Notice that the fluid also goes over here, and this is what we call the vestibule. The vestibule is the thing that deals with gravity and deals with acceleration. And then there's the three semicircular canals. Now, if you were designing the system for semicircular canals, how would you orient those three semicircular canals if you wanted to help you with direction and dizziness and all that jazz? Yeah, within different planes. And you'd probably want one plane to go this way, and you'd want one plane to go this way, and you'd want the third plane to go this way. So it goes this way, and this way. X, Y, Z planes, ever heard, remember that from your geometry back down the road? So anyway, that's how they're oriented, 1, 2, and 3. You don't have to learn which one's the anterior, the posterior, or the middle, or anything like that. Just know that there's 3 of them, and they go in, they're all at 90 degrees to each other. Okay. I'm going to come back and talk about that side over here. Let's talk about the cochlea over here a little bit. It's not going to let me. It's going to keep telling me to teach all 3 of them at the same time, so here we go. The vestibule. The vestibule includes 2 little things called the saccule and the utricle. Let's go back over here. And let's see if we can't find the saccule. Here's this little sac right here called the saccule. And here's this little other nook and cranny called the utricle right here. And these are the 2, the saccule and the utricle have receptors in them. They're called hair cells that provide sensations of gravity and linear acceleration. When I say linear acceleration, Katya, what does that mean? Okay. Kyle, what does linear acceleration mean?
Maybe acceleration in a straight line.
Yeah. Well, have you guys ever ridden in a race car or a fancy sports car? And you're sitting there against the seat, and the guy throws it into first gear, and you kind of get thrown back against the seat, and then off you go. Well, you realize that you're accelerating, that you're going really fast, and you're going forward, right? The reason you know that is because of this thing right here. It's telling you about linear acceleration. Now, if you've ever been to Disneyland, and you've gone on one of those loop-de-loop roller coasters, you know when you're upside down, don't you? You know you're upside down because your head, which is used to sitting like this, upright, where that's up and that's down, suddenly you find yourself upside down, and you can tell that, right? I mean, you can tell the difference. And the reason you can tell the difference is because of this as well. We know which way gravity is supposed to go, and if you go screwing that up, you go screwing this vestibule up, and you're going to probably feel dizzy or you're going to feel sick at your stomach because we're just not very used to it. The semicircular canals they contain the semicircular ducts, and they have receptors that are stimulated by rotation of your head. All right, let me tell you about the semicircular canals. Let's just assume, you guys ever have a hula hoop when you're kids? Okay, well, let's think about it like this. We've got a hula hoop, and it's plastic, and it's that size. And let's hold it here in our hands, okay? And the hula hoop has got water in it, okay? And the hula hoop has also got some sand in it. So there's sand in there in the water that's in the hula hoop, okay? And now we're going to get on the tilt-a-whirl, and we're going to start doing this. Okay? And then all of a sudden we're going to stop. Okay? What happens to the water in the hula hoop? The water keeps moving. How about the sand that was moving with you? As the water keeps moving, the sand is going to keep moving some too, right? And so even though the wall of the semicircular canal at first is moving in this direction, is the water really moving in that direction? Not so much. It's kind of inert, right? I mean, it's got inertia. It's going to stay there. So although the semicircular canal is moving this way, the water, the inside of the semicircular canal thinks, well, wait a minute, the water's going the other direction. Think about that, and I think that'll make sense to you. So the whole idea is that on the inside of this semicircular canal are these little hair cells that kind of stick out into the semicircular canal, and as that fluid moves past them, they're sitting there in the breeze or in the flow. And when the flow goes one way, the hair cells get bent that way, and that sends a message to your brain. And then when you stop and all the fluid flow starts going in the other direction for a few seconds, they get flipped back the other way, and that sends just the opposite. And that's why when you step off the tilt-a-whirl, you can't walk very good. Whoa, right? Because your brain has been getting all these screwed up messages. And people pay money for that. They like getting their brain screwed up. That's why we ride carnival rides. That's why I hate them. The cochlea, we're still trying to get to it. It's the cochlear duct. And the cochlear duct is nothing but a very long portion of the membranous labyrinth, but it's coiled up, okay? It goes up in a coil and comes back in a coil. That's so you can get it all in a small area, and it fits in there. The round window is a membranous branch at the end, where if the wave of motion of the fluid goes all the way to the top and comes all the way back down, it hits the round window, and it sort of bulges out into the middle ear, which remember is full of air, and that deadens the sound. So it doesn't then go back up again. I'm glad you could make it. We held the quiz for you. Thank you. You're welcome. The oval window is where the motion goes into the cochlear system. That's where the stapes fits. The stapes fits right on what's called the oval window and gets that fluid on the other side moving. Connected to the base of the stapes. Okay. Boy, it just won't let up, will it? Oh, it's going to make me teach all three of them all at the same time, as I said. Okay, so the sense of gravity and acceleration is sent to our brain from hair cells that are located in the walls of the vestibule, that little common area down there. The sense of rotation comes from the semicircular canals. They come from the hula hoops. And remember, there's 3 hula hoops. There's a hula hoop that goes in this direction, there's a hula hoop that goes in this direction, and there's a hula hoop that goes in this direction. All right? 3 of them at 90 degrees from each other. The sense of sound comes from the cochlea, and it depends on 2 things. It depends on the frequency, wah, 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 low tunes, wah, wah, high tunes, okay, different frequency. And it also comes from amplitude. What's amplitude mean?
It's the height of the wave, isn't it?
Yeah.
The
Amplitude is the difference between me saying and me saying same pitch, just different amplitude, right? Different pitch would be that's 2 pitches, 2 different speed of the vibrations. I remember when I was in college, everybody, it was so cool to buy a really good turntable. We didn't have CDs or tapes or any of that other stuff. So you bought vinyl records, you put a little needle on it, and it turned around at 33 1/3 times a minute. And it measured the vibrations off of the groove as it went around the record. And so when you went to the Radio Shack catalog to buy your stereo system, because everybody wanted to have the best stereo system, right? You know, you would buy the more expensive speakers would relate to lower frequencies down in the 17 to 15 range, and the better speakers would go way up high into the 40 or 50,000 cycles per second, the high notes. And it was an extra $100 to get that extended a little bit on both ends. So it would go down to say 12, and it would go up to 50,000 on the top end. We don't think about any of that stuff anymore because it's all digital, right? And no big deal. Boy, it was a big deal back then. Okay, let's talk about this equilibrium, this business about getting dizzy and stuff. And let's talk about these hair cells. I've mentioned them a couple of times. But basically, hair cells are the basic receptors of the inner ear. And they provide information about the direction and the strength of mechanical stimuli. Now remember, in the semicircular canals, that means which way is the fluid flowing in the hula hoop, right? In the vestibule, it's whether we're being forced forward really fast, the linear acceleration thing, or gravity is pulling us down. But once again, the reason we interpret it is because of the way those hair cells are getting pushed one way or the other. We'll talk about the cochlea in a few minutes. Okay, so the semicircular canals, the ducts, are continuous with the utricle, that little, the den sort of where everything connects into. And each of the ducts contains a little ampulla, which is where it starts, has a little gelatinous cupula. You should definitely see these things in the lab because we have great slides of these hair cells and cupula and the stereocilia, which kind of stick out the top, and they kind of stick up into this fluid so that as the fluid spins, the stereocilia kind of go one way or they go the other way depending on which way the fluid's moving. Some of them have a kinocilium, which is a single large cilium. No big deal there. So it kind of looks somewhat like this. Here's these 3 semicircular canals. Notice there's fluid that goes up into them and goes around like this. They go up into that one or they go up into this one. And this little thing right here is what really reads the flow. It tells you whether the flow is moving this way in that semicircular canal or whether it's coming this way. Same as with this one, is the one that tells you whether the fluid is going that way or this way, and same for this one, for the third one. The saccule, macula, I guess, right here, is what's going to help you with your gravity and linear acceleration. Okay? The 3 semicircular canals are just for rotational feelings when you're spinning. Keeps you from spinning. Here's another kind of a close-up thing of it, with the hair cells sticking up into the flow, the fluid going back and forth through there. Example of what I've been trying to say, when the duct is rotating one way and the fluid goes this way, then the hair cells get flipped over to the side, back and forth. And it's actually the direction that those things go which sends the signal to the brain. Okay, so these are mechanical receptors. They're measuring movement of fluid to get these symptoms to you. Close-up picture, sensory nerve ending down here. This is a hair cell, has these hairs coming out the top of it. Call them stereocilia, calling one of them kinocilium, I guess is fine. But basically, depending on which way the fluid goes determines which way the cells are stimulated. Now, when I told you the story about the hula hoop, and I said the hula hoop was full of fluid, and then I said you put a little sand in it, okay, because that way you've got even more to feel moving back and forth. Well, that's because that's the way it really works. You see, in addition to just being full of fluid, there's a bunch of these little otoliths, little pieces of sand, so to speak, that live in this fluid that get swept one way or the other. And when the little pieces of otoliths, also called statoconia here, get flipped back and forth across this gelatinous kind of stuff, it's purely just fluid, it stimulates it causes these hair cells to be flipped one way or another, and then the signal gets sent to your brain. So, that's why I told you about the sand in the hula hoop, because there really is sand in your inner ear. Okay? The gravity thing, the way it would be stimulated, if your head's in the neutral position, gravity would be pushing down on these hair cells. If you move back, then the little stones, the little rocks in there, the otoliths would move downhill. It would push the hair cells one way, and that would be the signal that would be sent to your brain. And that's why you would know that, whoa, wait a minute, my head is back. It's not neutral, it's not forward. Because these are the otoliths, these are the hair cells that got stimulated by you moving back like that.
How do the otoliths reset?
They're like sand in the hula hoop.
So then
They just keep moving around. They're always going to stay there. And it just depends on whatever they roll by is what they stimulate. Okay? Just laying there and not moving, they don't really stimulate much. We studied the brain, and we studied these tracks, that carry the nerve impulses back to the brain. And remember, this is cranial nerve number 8, and the axons go along the branch of the vestibulocochlear nerve, and they go back to, guess what? Vestibular nuclei. When you think dizziness, when you think motion, when you think orientation, when you think acceleration, think vestibular. Okay? That's kind of what this sense is, sense of vestibular sense. Notice there are 2 branches. Here's the vestibular branch coming from all of this stuff, the vestibule and the semicircular canals coming here. It meets up with the cochlear branch. The cochlear comes from the cochlea, which is not in the picture here, and it goes from there up to the brain stem, right? What is this? The vestibular nucleus, somewhere in the medulla, right? and then it gets passed around through the brain to different parts. Now, just don't memorize any of this stuff, but just so you think about it a little bit. So here's nerve 11. What does 11 go to? Right? So if you're getting spun around, you're trying to compensate for it, right, by moving. And so that's what all this information is being digested for. Here's nerve 6. What does 6 go to? Lateral rectus, right? Eye muscle. What is 4? Trochlear nerve goes to the superior oblique, right? Rotates your eyeball. So think about it. If you're on that tilt-a-whirl, you think your eyes are flashing around. Sure they are, because they're getting all these weird instructions that are coming in from this dizzy vestibular apparatus back here. It sends all these information up here, and your eyes are flipping around. And we call that what, when your eyes flip around in an abnormal way? Nystagmus, that's right. Nystagmus. Remember, that's the test that the cop does when he thinks you're drinking too much. He puts the light in your eyes, and he says, watch my finger. And you go over there to watch the finger, and meanwhile your eyes are going, boom, 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 boom. It's out of your control. Keep that in mind next time you're drinking. Eye movements. Our eyes always want to focus on a specific point. They do not want to be just wandering in space. And so the way we do that is when we're like, I always remember this when I was a kid, and we used to have merry-go-rounds in the playgrounds. I think they took them all out because people got hurt on them, I guess. But you'd be sitting on the wheel around, and your brother or your sister would get it going as fast as they can. And if they were on there with you, if you had someone else on there with you, you'd look over at them and you'd look at their eyes, and their eyes weren't just sitting still and going around. Your eyes are going toom, 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 because our eyes don't like to see blurry. They want to stay focused on something. So as you go around on that merry-go-round, your eyes are doing just that. It's a normal thing. Your eyes move from point to point to point to point so that it's always in focus, and our eyes can move pretty quickly. Nystagmus, we just mentioned, can be caused by damage to the brainstem caused by alcohol, which is a temporary thing, hopefully, or it can become from all these weird stimulations in the inner ear, and that's what occurs down at the carnival. You get spun around. When you get off the tilt-a-whirl or one of those spinning things, your eyes are going to be flipping around, let me tell you. Look at it next time. Watch your kids. Your kids do it a lot more than you. Let's talk about the cochlea for a minute. So this is the hearing thing. And once again, I said that the vibrations of the tympanic membrane go through the three ossicles. The last ossicle is the stapes. And the stapes vibrates, and it causes the fluid inside over here to vibrate up and down through the cochlear duct. Basically, the sound goes up through the tympanic duct, and then it goes to the top of the pointy thing up here, turns around, and comes back down through the cochlear. Let's see, what did I say? It goes up through the tympanic, down through the cochlear. Here's the key. Be sure you get that straight. Here's what it looks like under the microscope. This is a really tiny deal, just so you know. Your cochlea is pretty small, but here's the way it works. It comes up from the oval window, from this one to this one to this one, and then starts back down from this one to this one to this one. So it's going up and down, and all along the way, there are hair cells. And the way it works is the lower frequencies get dumped off earlier. So the lower frequencies are actually heard down here at the lower end, closer to the oval window, and only the higher frequencies get through up to the top and start back down on the other side. So we hear our low frequencies first as we're going up, and we hear our higher frequencies coming back down. They're the only ones that are still going, that haven't gotten damped out by then. This is what it looks like under the microscope. If you slice down through it, you're going to catch ducts going up and ducts going down. And in between the two, you're going to see this thing called the organ of Corti. And I just tell you, you don't need to look at this in the lab. It's really cool. The organ of Corti is what we really hear with. That's really where the hair cells are that interpret the sound based on the frequency and the amplitude. The amplitude tells us how loud it is. The frequency tells us the pitch. There's a couple of physics things you need to know here. I've just already mentioned them, really. The frequency of the sound is determined by which part of the cochlear duct is stimulated. Like I said, the lower ones are interpreted first, the higher ones are interpreted later. The intensity tells you how loud it is, and the loudness is determined by how many of the hair cells are stimulated. If there's just a few, it's probably a pretty soft noise. If there's a lot, it's a pretty loud noise. This is the organ of Corti. I'm just going to let you look at that under the microscope. Here's a nice diagram of it. You will see these. Now, let me give you one little heads up. If you look at a slide that looks like this, you're going to see, you would like to think you're going to see a perfect organs of Corti in every one of them. You won't. On the entire slide, you might see one organ of Corti that really looks like it's supposed to look, which is like this right here. Okay, so just keep that in mind. Don't get frustrated. Call me over. I'll look at it with you. Perilymph is in. It's on the way back. Yeah. So you see here, if this is an organ of Corti right here, you see there's nerves coming out of there, and those nerves are going to all grab together and join up to form the cochlear branch of 8. In other words, the hearing part of nerve 8. Cool looking. So what are pressure waves? Well, they're sine waves. You remember sine in trigonometry and math. And I think the better way to think of it is like the speaker, and the same as the tympanic membrane is vibrating back and forth, and so it's causing these little regions where the air molecules are crowded together, going through. The pressure waves, the wavelength is the distance between two adjacent pressure waves. As I said before, if it's a really low-pitched sound, they're going to be pretty far apart. If it's a really high-frequency sound, they're going to be just one right after another, real high-pitched. Sometimes we use the term cycles instead of waves. Another word is hertz. Hertz means the number of cycles per second. You don't have to say hertz. You don't have to put an S on hertz. It just means a multiple, plural number by itself. Cycles per second. Let's talk for just a second about amplitude, which is, in other words, how loud something is. Let's talk about decibels. Y'all have probably heard of that before. Any members of rock bands in the crowd? Wouldn't have mattered. You couldn't have heard me anyway because they've all had their ears destroyed. Because the amplitude of the sound up in where the band plays is so loud that they're suffering damage to their ears every minute. Most of them are wearing plugs these days or some kind of ear implant thing to keep from damaging their ears so much, but it's always been a problem. We can use tuning forks. We have tuning forks in the lab that you can cling and see the difference in wavelengths. This is the wavelength from one wave of energy till the next. And this, let's just go back to that thing I was mentioning to you about the cochlea. Did I tell you this wrong? Anyway, here's where the high frequencies are heard down to the lower frequencies. This looks like I did tell you wrong. The stapes, which comes in at the oval window. So the high frequencies are heard first. Told you wrong. You'd hear the higher frequencies up front, and it'd be on up around a couple loops before you get down here to where you're starting to hear the lower frequencies. I don't think I can really add anything to these little diagrams. You ought to read that part of your book. As far as how do we hear and how does it get to our brain? Well, here's the cochlear branch coming from the cochlea that's picking up all these sound messages. They're coming back here to the cochlear nucleus in the brainstem once again. They're going up here and working with these guys in the mesencephalon. Some of them are getting up to the thalamus, and if we think we ought to hear it, we do. If it's the fan and the projector, it doesn't make it all the way up, so we don't hear it. Our hearing range is amazing. Our hearing range is a trillion-fold. The sounds we can hear, the slightest sounds we can hear, and the loudest sounds we can stand are about 1 trillion times different from each other. Children have the greatest range of hearing. As we get older, we have hair cells and parts of our cochlea are dying off. You ought to know a little something about this. Decibels, if you have an OSHA, if you work on a construction site that has a lot of noise, when you get to up to about 80, you're going to have damage in your ears if you stay in that loud noise for up to 8 hours. And once you get above 80 to 90 decibels, 100 decibels, that's a chainsaw, boiler shop, pneumatic drills. The most you can tolerate is 2 hours. If you're at a heavy metal concert, it's 120 and it's immediate danger to your hearing. You just got to have ear protection on. Jet plane. Out on the aircraft carrier, when I was a young man in the Navy, there would be 15 jets running at the same time, all within a 5-acre piece of tarmac there. And you just cannot imagine how loud it was. But everybody had on Mickey Mouse ears, we call them, but they were ear protection because you just couldn't stand it. It was so loud out there. They never thought about putting a silencer on a Navy jet or anything to kind of quiet the noise. Rocket launching pad, hearing loss is inevitable, so don't go there. Real quickly, with age, your hearing gets worse. Your tympanic membrane doesn't vibrate as well as it should, so I can't hear some of you talking from the back of the room. The articulations between the ossicles start to stiffen, so it doesn't move the membrane as well as it should. The round window may begin to ossify and get hard with calcium in it. Hey, what did you say? Are we done? Oh, yeah. I took this picture about 6 or 7 years ago. I was out in Sherwood with the kids, and they were playing video games. And there was this old video game called X-Men. Y'all ever heard of that? It's probably from before your time. It was one of these stand-up things where you had guns, I think, and shot. Well, my uncle, William S. Sessions, used to be the director of the FBI. He was appointed by Reagan and fired by Clinton. He put signs like this on all the video games in America, thinking this will be the solution to the war on drugs. Winners don't use drugs. So it came up, and I took a picture of it. He's still alive. I haven't talked to him in a while. I communicated with him on Facebook a few times in the last year or so. But is this the way to win the war on drugs? How's it going? Are you farsighted or nearsighted? A couple of things that you might want to check out. There's a really good video from Khan Academy, K-A-H-A-N. It's called Auditory Structure, Parts 1 and 2. They're really pretty short. I think they're worthwhile. I have a couple of little videos that I'll show you over in the lab as well. Okay? Why don't we call it good? I passed everything out I can pass out. So maybe if you would pass those around. We'll just do the quiz. I think we can do it now. We've got a few minutes. All right, so that finishes up the chapter. I guess the thing I want you to realize is that when we talk about these 5 senses, the first 2 are chemoreceptors, right? They're chemical reactions that leads to you being able to smell and you being able to taste. For you to be able to see, it's a photochemical reaction. The light coming through changes the rhodopsin, remember, from cis to trans, and that eventually generates a electrical signal. So that's how your vision works. And then these last 2 combinations, and it's really more than 2, but hearing and the different vestibular activities are all mechanical. It's truly a mechanical system that allows you to hear and to feel gravity and to get dizzy. Thank you, sir.